this is a particular privilege to be here, and I'm so looking forward to meeting some colleagues from Australia and working together. How about we just go around the room, maybe Laura, you might want to introduce yourself next, and then we'll keep moving across my screen.
Hi everyone, my name's Laura Burks. I work with Geth and Thomas at MND Australia to administer our research grants program, and it's really great to see so many faces from lots of different backgrounds here today, and I'm really looking forward to getting to know all of you a bit more. So thank you, and I will pass on to whoever's next on my screen, if that's all right, Terry.
Yeah, that's fine.
I'll hand over to Rosalie is next on my screen.
Okay.
So. Just on mute, Rosalie.
Sorry, Rosalie, you said you're unmuted, but we still can't hear you. I don't know if it's on our end or your end.
Looks all good from this end.
Right. Perhaps while you sort that out, we'll move on to the next screen. Carol, you're next on mine anyway.
Thanks, Catherine. Thanks. Morning, everybody. I'm Carol Burks. I'm the former CEO of MND Australia. I worked with people living with MND for just over 20 years with MND New South Wales and then MND Australia. Also was chair of the International Alliance of ALS MND Associations for a few years. So I'm very excited to be part of this program and see it now being introduced into Australia. So thank you for having me.
Perhaps next, Benjamin. Benjamin?
Yeah, hi. So my name is Benjamin from GenieUs Genomics. So I've got three colleagues here, Durley, Shreya, and Cameron. And we are hosting this in partnership with MND Australia. We're very excited to be hosting the inaugural MND CRLI. We know we've got a brilliant 2 days of workshops for you guys to inform you, get you involved in the process. So I'm really looking forward to it. Any questions at all, you can refer to, as Gethin said, you can also come to myself, Shreya, Durley, or Cameron, and we're happy to help you with whatever. We just want you guys to feel comfortable and very involved. So yeah, looking forward to it.
And my colleague, Rick Bedlack.
Hey, thank you, THP. So my name is Rick Bedlack. I'm a neurologist. I run the Duke University ALS program. And within that program, we've got a big multidisciplinary clinic. We do a lot of different kinds of research. And I'm really excited about some of the patient advocacy that we do. This program, which we'll talk a lot more about in a few minutes, ALS Untangled. And out of that has grown a lot of interesting things, including the ALS Reversals Research Program. So it's great to be with you.
Thanks, Rick. Keith is next.
Hi, my background, I spent 32 years full-time in the Australian Navy, followed by 16 years shared between defence industry, shipbuilding industry, project management, and on the Naval Reserve, assisting with some engineering mentoring, I guess you'd call it. These days, I have a role in the board as a director of MND Association Queensland. I'm also assisting with the registry we call MND here in Australia. And I've been living with MND since 2015. Thank you.
Thank you, Keith. And next, Michael.
My name's Michael Turk. I've been retired from UWS for the last 15 years. and I was carer for my late wife for 2.5 years with MND until she passed. My research interests are in IT. I spent 20 years doing commercial IT and 20 years as a academic Western Sydney unit. My current interests are in using artificial neural networks to analyze data and things like that.
Thank you, Mike. Nice to meet you. Jojo?
Hi, everyone. My name is Jojo. I'm not a professional yet. I'm just a Master's of Nursing student. So looking forward to working with you guys.
Thank you, Jojo. Nice to meet you. Allison?
Hi, good morning, everybody. My name is Allison, and I have the pleasure of working with Dr. Bedlack and Dr. Hyman Patterson at NEALS. I'm the co-chair of the Patient Education and Advocacy Committee for the NEALS Consortium, and I'm also a surviving spouse of ALS, having lost my husband to the disease. So Michael, I feel you, I'm glad you're here. And Keith, I'm not sure if it's appropriate. I'm in the United States, of course, but just say thank you for your service. It sounds like you've had an amazing career. So thank you for being here.
Thanks, Allison. Good to see you. Karen?
Hi, I'm Karen Hutchinson. I'm a researcher with Macquarie University, and I'm also a physiotherapist. So I've worked many years with people living with MND and their families in the UK and in Australia. So lovely to be here and to meet you all.
Thank you. Nice to meet you. Anjali?
Good morning, everybody. So my name is Anjali Henders. I work at the University of Queensland, and I'm actually the program manager for the SALSA Systems Genomics Consortium. And that essentially brings researchers and clinicians from around Australia together, where we have this very collaborative project around collecting clinical data and biological samples for the purposes of progressing ALS genomics here in Australia. So it's nice to meet everyone and looking forward to the next couple of days. Thank you.
Thanks so much, Anjali. Colin?
Everybody, I'm Colin Mahoney. I'm a neurologist. I started my career more in the area of frontotemporal dementia, and did a neuroimaging PhD at UCL. Then I kind of came over to Australia, doing a postdoc at the University of Sydney, and got a lot of experience in MND and clinical trials. And now I'm actually based as a neurologist at Liverpool Hospital, supporting the MND clinic here. So that's about 20 kilometers from the centre of Sydney, not in the UK. And it's a pleasure to be here and contribute.
Thank you, Colin. Nice to meet you. Christine?
Good morning. I'm coming from Brisbane in Australia. My background is as a geographer degree, but I actually am one of the people that have lost a parent to MND, and I've also lost my uncle, and they both caught it at the same time, and they both passed away at the same time. So that's my experience.
Thank you so much. Jen?
Hi, everybody. My name is Jenny Hartman. My background is as a Master's Qualified Librarian. I've been in the broader information sector for the better part of 20-25 years, spanning publishing, library systems, library supply, working with researchers, clinicians in various capacities in different sectors. I was recently diagnosed. I was diagnosed 2 months ago. I have a young family. I've got 2 boys. Thank you.
Thank you. Casey?
Good morning. My background is in government relations and strategic advisory. I'm a partner at a firm here in Canberra where I'm based. My mum was diagnosed with MND a couple of years ago, and I don't really know I don't really have much more of a background other than via my mum, and I was just very interested to see what research and clinical trials were out there. So I thought I'd join this program. Thank you.
Thank you so much. Nice to meet you. Karen? Oh, sorry, Karen. Got you while you were drinking. But Karen? You have to unmute. Yeah.
Yeah, I've already introduced myself. Sorry.
No, I'm sorry. I'm like,
no, that's all right.
I know it's hard. There's a whole Peter. Peter, yeah, I mean.
My name is Peter Clark. My background is a cabinet maker, and I've had MND for nearly 10 years. I've participated in numerous trials and still am participating. So I thought I'd have a go at
Okay. I don't know. It's frozen here.
Yeah, I think Peter's just frozen a bit.
Okay. So I think, Rosalie, did you want to try again?
Still can't hear you, Rosalie. I'm going to just send you a little direct message. It'll pop up in your little chat down the bottom with a few ideas about why we might not be able to hear you. So we'll do the last few people and then maybe come to you. Can we go to Alan next while I message Rosalie?
Yeah, Alan.
Hi, I'm Alan McRae. So I'm a genomics researcher and with a focus on MND research. So I'm working with the SALSA consortium to generate big genomic data to understand the biology behind the disease and what makes everyone different in terms of disease progression.
That's great. Thank you so much, Alan. Nice to meet you. And then Shreya, I don't know if you're there. Yeah.
Hi everyone. Yeah. Yeah. Hi everyone. I'm Shreya from Genius Genomics. I just realized my camera's off. Sorry. Hi everyone. Yeah, I'm Shreya from Genius Genomics, just following on from what Benji said. If at any point anybody needs any help, we're happy to assist with whatever is needed during the workshops. Just let us know, and we're super excited to be hosting this event. Like, yeah. Thanks, Terry.
No, thank you. Cameron? I don't know if Cameron's there again. No, the camera's not on, so. Cameron, are you there? Yeah.
Sorry. Hey, I'm Cameron. I'm also from Genius Genomics on the research side. So yeah, most things have already been said by my colleagues, Benji and Shreya. Yeah, anything you want to know, just, yeah, let us know.
Nice to meet you. Matt? I'm not sure Matt's there. Darley, are you there?
Darley's just lost her voice. She's got a bit of a cold and flu.
Sorry
Yeah.
to hear that. Nice to meet you anyway.
Yeah, she's the marketing coordinator at Genius Genomics. She's helped organize this along with me, Benji. And yeah, if you need anything, just hit us up.
Yeah, and I don't know if Rosalie has made any progress with her microphone.
And Matt's on camera now as well.
Oh, hi, Matt.
I think he's just sorting out his, there you go.
Oh, he's driving.
Hi, everyone. Sorry, fingers don't work so well. My name's Matt. I've been living with MND since late last year. I've been involved in a lot of the research trials at University of Queensland with Rob Henderson and Derek. So I'm still involved in some trials, but my background is 30 years as a geochemist and qualifications in chemistry and management.
Thank you, Matt. It's very nice to meet you. And
Likewise.
thank you for coming. And thank all of our pals who are attending. Rosalie, have we made progress yet?
Yes. Can you hear me now?
Perfect.
All right. So basically, I'm a nurse, and a couple of years, in 2019, I completed a doctorate in nursing, and it was a qualitative research. So that's my expertise. But I am interested in motor neurone disease because I have my grandmother, and her two brothers, and my uncle. I have two uncles that have all died from motor neurone disease. So it's quite strong in the family. And so that's, I'm interested in continuing either doing further research or getting involved in certainly being able to help change perceptions in the community.
Well, thank you so much. Appreciate your being here. And who has not introduced themselves?
I think that's everyone, Terry.
Have we gotten everyone?
Yep.
So,
There's a few more people to join in. I think as they come in, we can sign them in.
well, I want to say it's really, again, a pleasure to meet everybody and appreciate the interest in the program and the support for the program. And we're going to hear from Rick now, Dr. Bedlack, on why this program is necessary. Rick will probably want, there we go.
All right. Can you all hear me?
Yep.
No? Yeah. Good. Okay. Well, hello again, everyone. What an incredibly talented and diverse group. I'm really looking forward to working with you. So I'm going to kick things off with this talk, why we need clinical research learning institutes. And really, the answer is right there on the first slide, advancing options for ALS through partnerships with people who are living with it. But I'll tell you how I came to this realization. So this is me back in the 1990s. And you can tell this picture came from that era for two reasons. One, I had the Jerry Seinfeld haircut. And two, this was the only time in my life when I ever wore blue blazers. I was in residency at Duke and I was just trying to keep a low profile and get through before I could really sort of let my freak flag fly with my crazy outfits. But it was during my residency when I came across ALS for the first time. And I think about that case every day because it changed my life. I was so amazed by that person's story, so amazed by the collection of physical findings I saw in their exam. I'd never seen anything like it before. And then I remember how horrified I was when my attending came in and said, well, this is called ALS. We don't know why it happens, and there's really nothing we can do about it. You just have to go home and get your affairs in order. And I kind of looked over at the patient and I thought, if I feel this bad about this conversation, I can't even imagine what this young man feels. And I drove home that day and I said, this is going to be the disease that I specialize in. I'm going to try to stay at Duke University and change the culture here, build a program, a clinical program to offer people options, and a research program to offer them some hope. So I traveled around the country and I got to spend time with people like Terry, took me under her wing, Merit Jakovitz, Jeremy Scheffner, Bob Brown, lots of great mentors. And I tried to take the best practices from their clinics and bring them back to Duke. So I built a big team, multidisciplinary team. We now have 20 different clinicians on our team, which you can see listed there. And, you know, we started to get a lot more patients. We started with probably a dozen people living with ALS around the year 2000 in all of Duke. And currently we have about 550 that we're following. And as we grew, we got to be able to start doing some research. And so again, thanks to people like Terry and Merritt, I got invited into some multi-center trials early in my career. And we've continued to be partners on those trials. And then I've also, as I mentioned earlier, developed my own unique research interests. And through all those programs, I can offer my patients some hope. And last but not least, I even put my own unique stamp on the clinic. I've always found that clothes are a way for me to maintain my positive energy and that they can rub off on others, put a smile on people's faces. And so once a month, I get my whole team to dress in some sort of theme. And we even invite the patients who are coming that day to join us. And it just takes the edge off what can be a very stressful day. And so after about 5 or 6 years of building this, I kind of looked back and I said, I feel like we're doing as well as anyone. I don't think there's anyone doing better than us. And then I noticed something that kind of surprised me. Only a very small fraction of the patients that I was following were enrolling in research studies. So in my clinic, it was about 8%. And I surveyed colleagues around the world, and it seemed like in most clinics, less than 10% of everyone with ALS was ever getting into a research study. And if you look at it a different way, we went back and did a literature search at all the clinical trials that have been published. And on average, those trials were enrolling about 2 patients per site per month, which is kind of surprising because most of the sites that do trials are pretty big and they might see 60 to 80 patients in a month and only 2 are enrolling in research studies. And then through that same literature review, I was surprised to see that even though the infrastructure had improved over the previous decades, it wasn't really changing the enrollment rate. So even though we were networked into groups like the Northeast ALS Consortium, the enrollment rate was staying about the same. And to me, it was a big problem because poor enrollment meant that my patients were being deprived of the benefits of being in trials. So in my opinion, the benefits include not only, getting to try something, which we don't know if it's going to work or not, but also getting the hope that comes from being in a trial, getting the altruism, the feeling that something good is coming out of this terrible thing that happened. And even there's some evidence from other fields that people who participate in research trials, no matter what they get, whether they get the experimental drug or the placebo, they do better medically than people who don't participate in trials. And no one knows for sure why that is. It might be the hope, it might be the altruism, it might just be that they're being seen more often. But either way, there clearly are benefits from being in trials. The other thing it meant was that our trials were taking longer. They were costing more. There were actually some examples of trials that had to be terminated without any definitive conclusion because they couldn't enroll. And even if a trial does fully enroll and finish and gets a positive result, if only a small fraction of the patients in our clinics are able to get into those trials, how do we know that the results can generalize? And we have this issue right now with a couple of drugs that are approved in the United States, Radicava and Relyvrio. They've really only been shown to work in a very small subset of patients with ALS. And so we hope they'll work in everyone, but we don't know for sure. And so to me, unless this problem could be solved, it was going to take us longer to find a cure for this disease than it should. And that just wasn't acceptable to me. So I got together with really smart colleagues like Terry, and I put together some surveys. So I surveyed my colleagues, and I also surveyed people with ALS, both in my clinic and also a larger group of people online. And I learned that this is a complicated problem. There's lots of reasons for it. One really obvious reason is there's just not enough options, especially back then. We've gotten a little bit more as far as options now, but back then there just weren't that many trials. Even this year in 2022, which I think was the biggest year we've ever had in the history of ALS, as far as research options, in the United States at sort of peak, there were about 2,000 spots open in all the clinical trials. And we have about 25,000 people living with the disease in our country. But the other thing I learned from these surveys is that there was a real disconnect. You know, we were asking patients to be subjects in our research. We didn't consider them to be partners. And I think as a result of that, a lot of patients were just not that interested. They didn't really understand research. They had a lot of misconceptions about trial designs, didn't really appreciate the models we were using, the kinds of effect sizes we were looking for, the reason why we were using inclusion, exclusion criteria, the reason we were using placebos. The trials that were being designed were very burdensome, lots of in-person visits where people had to travel from a long way and spend most of the day with us. And people were just generally frustrated by how long the whole thing took, especially back then, but even now, it can take years from the time that we open a trial until the time that we finish and get the results out there. And as a result of that frustration, a lot of people had just turned their back on trials and they had decided to self-experiment with what I call alternative and off-label treatments, vitamins, supplements, prescription meds that are being used off-label, things they found on the internet. And there was a lot of frustration in the patient community that doctors were not more interested in these kinds of products. And I'll always remember a quote that a patient said to me back then. He said, "Doc, we would be better together." And I took that to heart. And I started to look around at other fields to see how they were improving patient engagement. And I found this really cool program in Parkinson's disease called the Parkinson's Clinical Research Learning Institute. And what it was a crash course in all aspects of research that patients and families could attend. And when they finished with the course and graduated, they were called Parkinson's Research Advocates. And on a regular basis, the Parkinson's Disease Foundation would connect with the graduates of this program to make sure that they knew about all the opportunities they had in the Parkinson's space for influencing research. So I decided to get together with Terry and Merritt and Jeremy Scheffner, a whole bunch of colleagues from around the United States, and build ALS Clinical Research Learning Institutes. And I'm happy to say now we've actually held 23 of these. This will be number 24. Most of them have been in the United States. You can see the lists of all the cities and the dates. We had our first international one in Canada last year. And of course, this is our first one in Australia. And we've now trained more than 550 people living with ALS to be what we call ALS research ambassadors. And we don't just train people in this program and then let them loose. We actually connect regularly once a month with them through something that we call the PEACE Committee. PEACE stands for Patient Education and Advocacy Committee. And this is now run by Alison Boulat and Nadia Sethi, two people who have lived with ALS. And what we do is we invite all the research ambassadors to come onto a teleconference, and then we parade different stakeholders in front of them, people who have projects. They could be individual researchers like Terry and I. They could be sponsors like GenieUs or a pharmaceutical company. They could be patient advocacy groups or regulators. And the point of it is for them to present their projects to this group of research ambassadors and get a partnership with patients to make the research more attractive, more feasible, more likely to succeed. And I have to say, I've been blown away by what the previous graduates of this program have accomplished. They've taken their training and run with it in directions that I never imagined. So two of the guys from my very first Clinical Research Learning Institute, shown in these pictures, went on to start something that I'm sure you know of called the Ice Bucket Challenge, which in 2014 raised $200 million in one summer and resulted in an unprecedented amount of awareness of this disease. And these research ambassadors have been interacting with Congress in the United States. And as a result, they've helped to obtain and maintain pretty large pots of money for ALS research. So one is called the National ALS Registry. One is called the Department of Defense's ALS Research Program. Both of these programs now have over $100 million in funding toward ALS research to look back on. And then in 2021, they did something, they took it up a notch. They got Congress to pass something called the Act for ALS. And that was signed into law by our president, Joe Biden, in December of 2021. That's $500 million in new government funding for ALS research and expanded access. All of that is going to result in a lot more research opportunities for people living with this. And our ambassadors have partnered with us at our meetings. So for example, in 2016, a bunch of ALS researchers from around the world got together at a place called the CRLI House. And what we were trying to do is put together a best practice manual for doing clinical research. And we had research ambassadors there, and they made sure that certain things from the patient voice were included in this document that's out there. They wanted to make sure that we advertised our studies and educated patients in the right way. They wanted to make sure that we were offering compensation for the cost of travel. They wanted to make sure that we were notifying people who participated in research in a timely manner. In other words, they shouldn't be finding out about the results of research studies, from a magazine or social media. They should be the ones that hear about it first. Wanted to make sure that we were acknowledging all their efforts formally in our publications, that our publications would be in open access format, meaning that people living with ALS should not have to pay to read about the study they participated in. And they wanted to make sure they would be invited to participate in our scientific meetings. And for the most part, that is now happening in the United States. And they've partnered with individual researchers and companies. They've actually been reviewing grants, and they've been on several different patient advisory boards to help people like me and companies like MT Pharma, Biogen, Cytokinetics to change the designs of their trials to be more attractive and feasible for people with ALS. And they're out there spreading awareness of why it's important to participate in research. And they're doing this through speeches at support groups and fundraisers, through interviews with magazines and on TV. They're helping us to design better search engines that are easier for patients to use. And they're even just showing up in clinic to talk to newly diagnosed patients. And this is one of my favorite quotes by one of our ambassadors, Rick. He said, "I'm starting to see evidence that our efforts work. For the first time, our clinic's getting requests about trials from patients. I've been out speaking to support groups around the state, and it seems to have piqued their interest. And I'm working with my clinic. I'm making myself available on clinic days to provide information and guidance with the trial process." So that just blew me away, because I know it's not easy to come to ALS clinic. And here's Rick showing up in his clinic on a day when, he's not even scheduled to be seen, and just hanging out to make himself available to advertise the importance of research and to answer questions to patients who are there that day. And, they're also helping us to clear up misconceptions. Many of you probably know now more than ever that you can't believe everything you read on the internet. And we want to make sure that these folks are kind of, monitoring some of the larger chat rooms and Facebook groups to be sure that any kinds of myths and rumors that are in there, have some oversight. And they've come back now and they're teaching in the Clinical Research Learning Institute. So Allison and Nadia were both graduates from one of our previous classes. They are research ambassadors who've come back to participate in the program. They've started some new projects that I never envisioned. So Allison is involved in some podcasts, actually Nadia too, where they interview the author of a recent publication. So Allison does all the ALS Untangled papers, and Nadia does a lot of different kinds of papers that come out of the CREATE consortium. And the hope is that we can make science more interesting and understandable to non-scientists. And they've helped out with a good website, which is called Your ALS Guide. This website has tremendous amounts of really useful education about different clinical options for ALS, including short videos about things like feeding tubes and non-invasive ventilation and tracheostomy, et cetera. And they've even helped me out with this ALS Untangled program that I mentioned earlier. And so if you haven't heard of this, it's a program that I created back in 2009 to try and help patients and families make more informed decisions about these products that I call AOTs. And, there's 3 parts to it. You can read a lot more about it on the website, www.alsuntangled.com. So our ambassadors have helped me to optimize the website, streamline the program. They keep giving me new ideas for reviews. They keep giving me ideas about how I can make the information that we put on the website more accessible. And we now have these podcasts, as I mentioned, and we have translations into Spanish and Italian because of the ideas of research ambassadors. And finally, and maybe just, most amazingly, they've actually been helping to accelerate the regulatory pathway in Canada and in the United States. So here in the United States, they participated with many different patients and patient advocacy groups to create a document that the FDA puts out there on their website, basically a roadmap to how we think about drug approvals in ALS. And that might not sound earth-shattering, but for a long time, there was nothing like that. It was kind of a mystery how you got drugs approved in this country. And they actually participated in a whole series of hearings in front of our Congress and in front of our FDA. And the net result of those hearings is that over the course of this past year, our FDA went from, we're not even going to look at this new drug, Relyvrio, until there's a second clinical trial, which we expect to take 2 to 3 years, to the point where in September, they approved the drug. That's amazing. I never imagined that these ambassadors could do that, and they did. So in conclusion, what I've learned and what I'm trying to accomplish in the better part of the rest of my career is that it's really important to empower people living with ALS to be partners, not subjects. And by doing that, we've got so many more and better ALS options. And I just think back to that patient, and he was right. We really are better together. So just in summary, I'll just tell you about some expectations that we have, some things you can expect from the faculty. I think everybody here feels like I do. We're here to partner with you. So we're going to do some teaching, teach you about the research process, how to critique a publication, show you how exciting the ALS pipeline is, show you where some of the barriers to success might be, and how you can help us use education and advocacy to overcome those. We will set up monthly peace committee calls and meetings, and you will be invited. You can come on there and partner with different stakeholders to make research better. And we're going to be available to you. Now that you're research ambassadors, feel free to reach out to us if you're trying to get some ideas, if you have an idea for a project and you need some traction, you need some connections, reach out to us. We're happy to try to help. But we do have some expectations from you as well. We want you to be engaged. It's a little harder when it's virtual, but please, try to put those questions in the chat room in between lectures. Feel free to unmute and ask questions. And if there's something that you don't understand or something that bothers you, let's get it out there, because we learn from you in these programs too. We're going to do a research paper critique. It's really much more useful if you read it and try to critique it yourself first. And then, we can help you with the lecture to show, where you might have gone awry. But if you don't read it, it's not going to sink in nearly as much. When you're finished with the program, be sure to evaluate it. What we're doing over the next 2 days, it really doesn't look that much like what we did, 10 years ago. That's because each time we tweak this program based on the evaluations that we get back. And, embrace the training. When you're finished, go out there and be research ambassadors. Don't be afraid to take chances to improve ALS research. And, whenever you do something that's related to your training, your advocacy, be sure you go onto the Northeast ALS Consortium website and use the tracker to keep track of what you did. And the reason why that's important is because, these programs, they do cost money. Virtual ones are much cheaper than live ones, but they all cost money. And so whenever we want to start another one of these and we reach out to a sponsor, the first thing they do is say, can you prove to us that the people that get this training are actually out doing anything that's helping research? And the tracker is numbers that actually are real easy for them to understand. How many different talks have they given? How many different contacts with Congress? How many interviews? How many blog posts? So let's keep track of all that stuff together. So I just want to say thanks to a lot of different people, most importantly, the people living with ALS. I know it's not easy. I know it's not easy to put yourself out there with this disease. All I can say is that you inspire us. And that's why, you know, that's why we're here. I mean, you know, it's 6 o'clock at night and I literally raced over here from clinic to get on this program. And I think, you know, Terry feels the same way. In fact, I learned this from Terry. We're just so passionate because you inspire us so much. So thank you for that. Thanks to all the research ambassadors, Allison, Nadia, if you're with us, you know, you continue to amaze me with the directions that you take this. Thanks to MND Australia and GenieUs for sponsoring, and thanks to all the other faculty members that are volunteering their time. And I'll shut my screen down now and be happy to open it up for questions.
Thank you, Rick. That was fantastic and really puts it in context really well. And I can recommend the Create podcast as well. I listened to it myself and it's fantastic, nice small doses of really relevant information about MND ALS and some of the more interesting left of field treatments out there, which could hopefully save you some time sometimes as well. Do we have any questions? Feel free just to pop them in the chat if you don't feel like asking them up front, or we can always come back to them a bit later as well during the sessions, depending how people want to run it. We're a little behind the program, but that was probably quite ambitious anyway. So we can always, like I said, come back to a few questions later as they come up to. If we've got no burning questions, I can't see anyone with a hand up or anything. I'll just run into my talk and I'll probably rattle through this pretty quick, and so we can get into some more discussions. So I'll just share my screen as well. Just bear with me for a sec. Right, does that come up as a screen share? Okay, great. So I'll just give you a quick overview of basically the MND Research Australia sort of ecosystem space we work in. So a quick introduction. So MND Research Australia, who me and Laura manage, is the research arm of MND Australia, and we were established back in 1984 as a national organisation to promote medical scientific research into MND. And we actually amalgamated with MND Australia in 2010. Look, the research challenges that we all I think all researchers around the world can be divided into 4 basic areas, and this is the same for Australia. I think the US, Europe, it's all the same. Basically, we really still want to understand the causes and underlying biology that contribute to MND ALS. How a big problem is also how can we better measure disease progression and responses to treatment. Also a better diagnosis there as well. We need obviously to develop and test new treatments. And while we're working all this out, while we're still trying to measure the disease and develop those new treatments, we still have to make sure we look after all the people living with MND. So we still need to enhance that clinical and healthcare research to make sure we're providing the best possible care and the best quality of life that we can for those living with MND. And these research areas, they're incredibly broad, right from environmental factors, epidemiology. We'll hear more about genetics today. There's immunology. We all hear about neuroinflammation, metabolism, you know, factors such as hypermetabolism, why people lose weight. A lot about social research, also clinical research. We'll hear a lot more about that today as well. Cognitive studies. There's an incredible array of research going on in Australia around the world. We use cells, we use animals, as well as samples from patients as well to try and speed along this research. Within Australia, we're actually really quite blessed with our research resources. Australia bats well above its weight within the world, considering the size of the population. And a lot of that is due to a number of really quite effective funding organisations we have to provide funding for research. So we have us, MND Research Australia. I said we've been around quite a while. And since about 2015, Fight MND have come on the scene, and that's come out of Neil Danaher, who I'm sure you've heard of, and his family. The Danahers are a very famous Australian football family, and they've created this really strong movement to raise awareness of MND and also fundraise, mostly through their connections in the AFL. And they've really upped the game in the funding, amount of funding coming to research in Australia. In Queensland, we have MND, are an organisation that provide care but also fund research. Most of their funds come through us. And over in WA, we have Racing for Mindy, and they're an organisation that funds genomics-focused research over in WA. And we're also fortunate to have some government funding programs. We have the National Health and Medical Research Council, the NHMRC, and the Medical Research Future Fund. These are two large pots of money that fund research right across the medical research breadth, all diseases. And unfortunately, they only have about a 10% success rate. So, and only some of that will be going to MND, but it is available. Whereas you'll probably find us and Fight MND, we probably fund about 30% of the grant applications we receive. So we have a more successful funding rate, but we don't have the resources, obviously, of the government. And we also receive funding from Department of Health as well. We do sometimes for certain grant programs. What sort of programs do we fund? So project grants are a main one, where basically someone says, I want to actually undertake this specific project. Us and Fight MND, we fund small proof of concept projects. It's like, this is an idea, I'm not sure if it's going to work. Therefore, we'll say, okay, here's a bit of money for a year or two to you work that one out. Larger projects, the NHMRC fight those, the MRFF fund those. Fight MND have a larger program, and there might be 3 or 4 year projects that really take an idea that's been proven to work, and they take it forward to really establish whether this, what is the answer to the research question we're trying to ask. And that can be basic research to try and work out, does this gene contribute to MND, or right through we're testing this different model of care, does this actually work when we put it into the clinic. Clinical trials are also funded in Australia. A lot of these are funded by the pharma companies that develop the drug, but the NHMRC and the MRFF can also fund clinical trials, and so do Fight MND. They've contributed to a number of clinical trials in Australia. We fund fellowships. That's funding the people to do the research. We provide their salaries so they can focus on their research, and they and all the organisations basically fund fellowships. And there's large-scale partnership program grants as well, where we've all contributed to those, where basically national programs to really build infrastructure and resources to help research move forward. How do we decide who gets the money? It's a pretty well-established program that works pretty much across all the funding agencies. Basically, we put out a call for applications. We decide what sort of applications we want, so we have quite specific criteria. And we also tell the applicants what we're going to score. What's important to us? Is it how your experience in MND research? Is it how good is the project? How innovative is the project? What is the benefit, potential benefit for the MND community? And then we ask experts to review this. This is called peer review. So basically, we find out who else would know a lot about this research area, and ask them to give us their opinion of this grant, and they give us a score basically, and say this is what we think of it out of 10, out of 7, whatever our scoring rubric is. And then we get together with all the experts in a room together, and we discuss about who should get the money. In the end, we look at all the scores and discussion. We know we can fund, say, 1/3 of the grants, therefore which of those grants should get funded. But it doesn't end there. We also want to know how your grant's going. We don't just give you the money and then say, right, talk to us in 4 years and we'll see how it's gone. We ask for progress. We have 6 monthly progress reporting. Every 6 months, you've got to tell us how the project's going. And then at the end of the project, we do want to know what the outcomes were, what came out of it, what's the potential for translation, how is this going to go into the population, you know, what difference will it make, and you know, who have you told about this research? Have you engaged with the MND community, etc.? And you know, who knows about the results of this research? So these are all the things we really want to hear once we give people funding. There's a couple of really important joint initiatives in Australia as well that are really going right at national initiatives I just want to briefly tell you about. The first is the MND partnership. This is where we've brought together the Australian MND registry. Keith is involved with this quite strongly. And the sporadic ALS Australian Systems Genomics Consortium, which is a mouthful, but is much better known as SALSA. So this is a fairly unique approach where they brought together this registry and a genetics database. And this is funded by one of those large NHMRC partnership grants I talked about. And they collect clinical data, genetic data, they've got biosamples. And a unique feature of this is, as well as when you go to your clinic, your data can get entered into the database, but you can also enter it yourself as a patient or your carer. So it really allows us to follow people longitudinally and follow how their disease progresses. There's also going to be a portal to allow carers to input their data as well, so we can really make sure we're trying to care for the carer as well, which is something that perhaps is a little neglected sometimes. And this has a purpose-built web portal. And look, I encourage you all, and I'm sure you'll hear this about several times today and tomorrow, that you should sign up if you get the opportunity for the MNDs, for the registry. It's an opt-in. You have to sign up yourself. We can't sign you up without your consent, so you have to opt into it. Benefits of these registries: it gives us better clinical trial access. Companies want to come to Australia because they know that you know, we've got the population in one place. They know who is going to be eligible for their trials, and it really gives them a good idea about sort of basically what they're getting into. The data that comes from this will help drive healthcare policy, inform healthcare policy. It empowers the patients themselves to see how your disease, how you can better manage your disease. And as I said, yeah, it's an attractive pharma. And this is a truly national partnership. All the state associations are involved, all the large multidisciplinary clinics. MND Australia, MND RA, Fight MND, and MND-ME are all helping fund this. And we have a number of researchers involved as well, right across the breadth: clinical researchers, genetics, healthcare and social sciences. It's really, and this is another way as graduates of this program, you can really get involved and also make sure you go out and spruik MNDs through your networks. Another large network is the Collective. This is something us and Fight MND have put together. And what we want to do is to bring all the researchers around Australia together so we can make sure we're collaborating and sharing resources. So it's really to avoid duplicating and siloing. We don't want people doing these little research projects with no one else knowing what they're doing when someone might be doing the same project elsewhere. So, and really core to this is a lived experience voice. We have a management board with half the members of that board are people with lived experience. We really want to make sure that what the Collective drives is really going to benefit the MND community. I'll just flick through these couple of slides just to keep it quick. Research we have in Australia, just a couple of examples that are large-scale platforms. We have Brad Turner at the Florey Institute, who has a large drug screening platform. They take cells from patients and then grow these cells up, and then they look at different drugs treat. They've got a robotic setup so they can do high throughput screening and test thousands of different compounds to see what effect they're having on the cells. Mary Louise Rogers at Flinders University in South Australia has a biomarker facility where she's collecting lots of biological samples to identify new markers, new ways to measure the disease. Similarly, at Macquarie University, they have a large biobank as well, and they're looking at genetic factors, and they have researchers who can then take these genetic factors and work out what are they doing, how they might affect disease. Up in Queensland, I think Matt White said he's working with Derek Stein. He has a large research project looking at metabolic changes in MND. Why do you lose weight? Why you've got hypermetabolism? Justin Yerbury at the University of Wollongong, who himself is an MND patient, has trying to work out why your proteins misfold in MND. Why is this protein accumulation? Why is it such a problem? And then down in Tasmania, Anna King, Tracy Dixon, Kathy Blissett all work on the cell biology, excitotoxicity. Why are your neurons firing too much? And why is everyone different? Why all your neurons behave differently? On the care side, we obviously MindOz, we talked about. David Berlowitz in Melbourne is an expert in non-invasive ventilation. Susan Mathers has a great project on health literacy where they're looking at how you can better understand the information that's presented to you. Anne Hogden at the University of New South Wales has developed tools to help you make those really critical decisions, you know, such as do you need a feeding tube, etc. And Samar Un over in Perth is developed the Compassionate Communities Project, which is really bringing those difficult end-of-life conversations more into the open, out of the shadows, to really make it part of the community and not something people really don't want to talk about. So that's just a real quick fly through the Australian research scene, just to really try and set the scene a little bit and give people an idea of what research is happening and what we can do. Please, if anyone has some questions, if not, then we can move into some breakout rooms, I think, to let people just talk about their own experiences with research. A few people mentioned they've had some experiences, but it'd be great for people just to talk about them and share those experiences. Does anyone have any burning questions, comments on mine or Rick's talk?
I just wanted to say congratulations, Gethin. That's an amazing portfolio that you've put together there.
Yeah, well, like I said, I think Australia, we were very fortunate to have, we've been around a while, but Fight MND have really, you know, energized it with some significant, I think, between us, I think Australia is probably the second or third highest MND funded country, research funded country in the world now, considering we're only 25 million people. I'm pretty proud of that.
It's great work.
Okay, so I think next on the schedule was to move into breakout rooms. Laura, did you get a chance to divide everyone?
I have. I've divided everyone up. What I might do for simplicity's sake is I will stop the recording, keep half of us in here and send half of us to a breakout room, just so we're divided into two. Gethin, just a question about the schedule. For the networking break at 11, did you want us to remain in those breakout rooms or should I bring everybody back in at that point?
I'll bring everyone back in so then people can run off for any biological needs and coffees, etc., food, and then come back on camera when they're back on screen, and then just everyone can just have a random chat while they're here.
So we'll run the breakout rooms till 11 or shortly after and then bring everyone back in. Is that about right?
That sounds perfect. Thank you, Laura.
Okay, I'm going to send half of you off to another room now. And I'm
find out. Okay, so the next talk, we have, like I said, a really important aspect of research and your involvement in research studies, and probably is about ethics and informed consent. So Anjali Henders from the University of Queensland is going to give us a bit of an overview of that. So please take it away, Anjali.
Thank you very much. Can everyone see the slide set?
Yep, you're right up there. It's all good. And we can hear
Great.
you.
Thank you. So firstly, thank you very much to the organisers of this event. And it's really nice to be involved in this inaugural session here in Australia. So Gethin has asked me to present on ethics and informed consent and really how this sits within the Australian landscape. But before doing a really deep dive into the specifics of informed consent, I really wanted to provide you with some background as to why this component of undertaking research is really important in conducting human research. So I'll also give you a little bit of a background as to how I'm involved in MND research in Australia, take you through our national ethical guidelines, the process of obtaining ethical approval to undertake research, and some key elements of informed consent. And finally, just some reflections on how these guidelines are incorporated into the research protocols at the coalface of what we do. Has that changed? That's not changing. There we go. So as Gathan said, my name is Anjali Henders and I'm the Program Manager for the Sporadic ALS Australia Systems Genomics Consortium. I'm also the Project Manager for the Program in Complex Trait Genomics here at the Institute for Molecular Biosciences at the University of Queensland. And I've been involved in MND research now for the past 7 years as part of Professor Naomi Ray's research program into complex traits of psychiatric and neurodegenerative disorders. And so the vision of SALSA, and I'll just refer to it as SALSA now, was to build a resource that would really try and support the understanding the complexity that is sporadic MND. And to do this, we needed to be systematically recording the complexity of disease progression by collecting biological samples along with detailed phenotype information. And there are already efforts being made to maintain harmonisation with international consortia and clinical standards, but it was really important to have a biological resource that accurately reflected the cohort of MND cases that we had here in Australia. And so we have around 7 clinics, as you can see in the map, that are located across the country. And to date, we have clinical data on over 1,000 MND cases, biological samples from over 900 of those, and we've generated genetic information on 965. So the programs that have been going for about 7 years, and we're very much looking forward to continuing our recruitment of cases and controls into this program to really establish a really vital and important asset for Australia. So human research is conducted with or about people, their data and their tissues. And it's really about taking part in surveys or being observed by researchers, perhaps providing a biological sample, having information collected about you that might be in a repository or a database, or analysing data that's been collected or generated historically. And so the crucial feature about research ethics is to understand that in order to carry out ethically justified research, we have to be particularly aware of where the imbalances lie between the researchers and the participants and what might be best done to avoid ethical conflict. Much of the ethical conflict is based in the tension that arises between the researchers' concerns for the interests of the subjects on one hand and the interests of science, society and the future patients on the other. And so ethical research is more than just doing the right thing. It really is an ethos that permeates human research processes. And so interaction involved in human research has ethical dimensions, and it involves acting in the right spirit out of an abiding respect and concern for one's fellow creatures. But it would be remiss of me not to mention some of the unfortunate unethical experiments that have happened in modern history, where significant ethical issues were not reported or detected until well after these projects had finished. And so the Tzutzki syphilis study that left infected men untreated so that the natural course of disease could be observed, or twin experiments where separation at birth took place to understand how our identities are defined by our nature and our nurture. And of course, the human research experiments that were conducted during the Second World War. So just to give you a little bit of a timeline of the human ethics documents and guidelines that are in place. So the atrocities that were committed by the Nazi scientists in the name of research were investigated in War Crimes Tribunal at Nuremberg. A number of doctors and administrators were found guilty of systemic torture, mutilation and killing of prisoners while undertaking human research experiments. And so this led to the development of the Nuremberg Code in 1947. And these principles were further detailed in the Declaration of Helsinki, which was adopted at the World Medical Association in 1964. And most of these principles are still active today and included in guidelines such as voluntary participation, use of appropriately trained staff, and evaluating the risk versus benefit in the outcomes of any research. And Australia worked under the Helsinki Declaration until 1966, when it then developed its own, and it would then be another 33 years later before the next reiteration or the comprehensive statement was released in 1999. And this statement in particular included new sections 15 and 16, on the use of human tissue and human genetic research. And it was the writing of these chapters that was really my foray into human ethics, as I was fortunate enough to be working with Sister Regis at the Mater Hospital here in Brisbane, who was actually the lead for these chapters of the national statement. And so I had come from a background in forensics and working in the forensic morgue. So it meant that I had a unique perspective on the collection of human tissues and their use in research. So the NHMRC statement, which is the National Health and Medical Research Council here in Australia, was then substantially revised again in 2007 and again in 2018. And it's a dynamic set of guidelines that really responds to today's research environment, evolving technologies, and certainly public engagement with research. So as I said, the NHMRC has this set of national guidelines, and it fulfills their statutory obligation to issue ethical guidelines on research involving humans. It provides guidelines for all human research, and conforming to these guidelines is now part of institutional policy and certainly of all of our funding agreements that we have in place. It doesn't have a legal status in itself, but it does have legal status by reference to the Therapeutic Goods Act, which I guess is similar to the FDA system in the US. So it does have its limitations. Many of these guidelines do. So compliance with legal obligations is not within its scope. So for example, applications of the guidelines to clinical trials are not legally enforceable under those guidelines. And it is the responsibility of the institution and the researchers to be aware of any relevant legal obligations in undertaking the research that they're doing. So there are 4 principles of ethical research practice, and I just want to take the time to go through these with you now, because these really form the basis of the guidelines, but also the basis of the research that's actually undertaken and approved here in Australia. So the first is research merit and integrity, and research that has merit is justifiable by its potential benefit. It uses appropriate methods to achieve its aims, and it uses current knowledge in the field. And most importantly, the respect for participants is not compromised by the research aims. So researchers with integrity have commitment to follow recognised principles of research conduct, to conduct their research openly and honestly, and disseminate and communicate results to permit public scrutiny and to add public knowledge and understanding. So the second principle is justice, and researchers should make every effort to ensure a fair distribution of risks and benefits, and that there is fair access to the benefits of research. The process of recruiting is fair, and vulnerable groups should not be disproportionately bear the risks of research. So there should be no unfair burden of participation in research on certain groups. And most importantly, the research outcomes must be accessible to the participants. The third value or principle is benefits, and this is the likely benefit to participants must justify the risks of harm or discomfort to those involved in research. The researchers have a responsibility to design research to minimise the risks of harm and discomfort, and to really clearly articulate the potential benefits and risks for the welfare of participants within the research context. The final value is one of respect, and this means recognising the value of human autonomy and dignity. Participants have the right to an informed consent, respect for privacy, confidentiality, and of course, cultural sensitivities in participating in projects, and means recognising the value of human autonomy. So this ensures that individuals' dignity is valued and really to empower participants to make free decisions about their participation. So all of these values apply to human research and provide this kind of organisational context for the national statement. So a document that goes hand in hand with the national statement is the Australian Code for the Responsible Conduct of Research. And it has an essential role in promoting responsible research practices and good research governance. It promotes integrity in research for researchers and explains what's expected of researchers by the community. And it also provides information for researchers, institutions, communities, and funding bodies to manage situations where there's been a departure from the best practice guidelines. And by that, I mean where there is questions around misconduct in research or a miscommunication. A strong research culture will also demonstrate honesty and integrity, respect for human research, participants, animals and the environment, a good use of public resources or good stewardship of public resources and public funding to conduct their research, appropriate acknowledgement of the role of others in the research outcomes, and of course, as I've said, being responsible and open communication of research results. So we have these two documents and we have these 4 pillars that we need to work under. But what is the process of actually obtaining approval to undertake that research at your institution or your location? So there are really 3 stages of obtaining ethical approval. And the first is the preparation of the study proposal. And this includes a detailed description of the research project, development of the research tools, and by that I mean it could be developing online questionnaires, it could be developing diagnostic instruments or validating perhaps physical measures, and of course the consent forms, and I'll come to that a little bit later in my talk. Once this is prepared, it is submitted to the Human Ethics Research Committee, which is we call the HREC, which will then undertake a scientific and ethical review. And this can be done either in as separate stages or as one process. The scientific review is usually undertaken by scientific peers who form part of the prescribed complement of ethics committees, who evaluate the research design methods and that they are appropriate for the research that you want to undertake, that they're current, and that the power of the power of the study. And so this will this study provide statistically significant results to support the research questions. And finally, the ethical review. It's an evaluation of the project against the 4 ethical values, translating that to determine whether the overall risk of the project and if there are specific components of the national statement that need to be invoked. The review process is often reiterative, with the proposal being cycled back to the researchers and the committee to ensure all aspects of the guidelines are considered, and where perhaps poorly conceived or incomplete research plans can be amended. And I know this can sometimes be incredibly frustrating for researchers who just want to get on it and do their project, but it really does ensure that the highest quality of research is being undertaken, upholding the value of the participants' contribution, and certainly meeting the requirements for the Responsible Code of Research Conduct. So the level of review is primarily determined by the risks involved in the research, and specifically the kinds of harm, whether it's discomfort or inconvenience, or certainly greater than that, the likelihood of these harms occurring, and the severity of the harm in the event that they actually do occur. So based on the review of the risks or the harms, the national statement defines 2 levels of research based on risk. So a negligible or low risk research and a high risk research. So a negligible research, this is research in which there is no foreseeable risk of harm or discomfort, and any foreseeable risk is really no more than an inconvenience. And in low risk, the risk in which the only foreseeable risk is one of discomfort, and everything else is considered high risk research. In many cases, projects deemed to be negligible or low risk do not require a full ethical review by the committee, but instead are sometimes managed under a reduced committee or is approved by the chair themselves. So there are projects, however, that irrespective of the risk always require full ethical review. And I just want to highlight some of these, as these are quite specific to MND research and certainly research in Australia. So when we look at the national statement, there are several chapters that identify specific situations where projects must undergo high-level review. And I just want to point out these 5 that are highlighted here in red. So these are people who are highly dependent on medical care or who may be unable to provide their own consent, people with a cognitive impairment, an intellectual disability or a mental illness, research involving our First Nations people, interventions and therapies, including clinical and non-clinical trials and innovations, and human genetics. So as these subjects are less able to protect their own interests adequately, safeguards must also be much more stringent and wide-reaching than might be the case for research involving less vulnerable research subjects. And so when submitting projects that involve these particular groups, special consideration must be made in the study protocol as to how any additional risk or harm is going to be managed within that project. So now I want to talk to you a little bit about the research study protocol, and this really is the main document that is submitted to the Ethics Committee for review. So a clinical research protocol is a formal design of an experiment. It's the plan submitted to a HREC for review, and in some cases to a funding organisation for research support. The protocol usually includes a description of the research design to be employed, the eligibility requirements for prospective participants, the treatment regime, and the proposed methods of analysis that will be performed on the collected data. It's a comprehensive document. It's often very long, 20, 30 pages long, but really does provide the committee with all the relevant information they need to make informed decisions about the research protocol. And it's the quality of this document that gives the HREC confidence in the research that's being proposed, and that the appropriate and due consideration has been given to the individuals who may choose to participate in this particular project. And so what is informed consent? And informed consent is one of the founding principles of ethical research. Its intent is that human participants can enter research freely with full information about what it means for them to take part, and that they give consent before they actually enter the research. There must be no undue influence on participants to consent, and the minimum requirements for consent to be conformed is that the participant really understands every aspect of the research that they are consenting to. And so there are 2 distinct stages to obtaining informed consent for participants, and this also would apply for clinical trials. The first is giving information. The person reflects on the information that they are given, and they're certainly under no pressure to respond to the researcher immediately. And secondly, actually obtaining consent, where we as researchers reiterate the terms of the research, often as separate bullet points or clauses in the document, and the person agrees to each term, giving explicit consent before agreeing to take part in the project as a whole. So as broad as the realm of human research is, so are the methods used to collect informed consent, bearing in mind some projects, or in some circumstances, consent is not required. It's always dependent on the level of risk of the project and how the consent process will be managed within the study protocol. So I'm just going to quickly take you through some of the different types of consent. So the first one is implied consent. And this is often used in online surveys or filling in paper questionnaires, which simply by completing the questionnaire or the survey, you've implied that you're happy to provide that information and be involved in that particular research. Written consent is still probably the gold standard, and this is where time is taken by the research team to meet with the participant in the clinic or in a research environment, and they go through very systematically each component of the project, what the project involves, and what they would be consenting to before actually physically signing a consent form and having that witnessed by the researcher and date stamped. verbal consent, and we've heard in our breakout session that sometimes it's really difficult for people to actually attend clinic or research institutions to participate in research. But this is becoming, I think there's a lot more flexibility now in terms of the research protocols that are being approved, that we can now actually collect verbal consent. So it would be a case of sending a participant via email or through the post a copy of that written consent. Someone would still go through that consent form with them, and their consent to the project would be recorded and stored as a sound file for the purposes of auditing. Online consent has become very popular. And again, this would just be a case of a person agreeing to participate in a project simply by ticking a box or endorsing a question, yes or no, that they're happy to proceed. It's a very quick and very effective way of recruiting people into projects. E-consent, and this certainly has become, I guess, much more populous in the post-COVID pandemic, where we had to maintain social distancing and people weren't actually coming into clinics, or people were participating via telehealth. And this is where we've taken the written consent, put it into an online format that is quite interactive. So it can include videos, it can include recordings of researchers talking, and it takes the participant through each section of the consent form in the same way that we would for a written consent. But at the end of each section, the participant would need to agree to a statement. They sometimes need to answer a question. And by completing each section, it reassures the researcher that the participant has had the time to walk through the whole of the consent form with an understanding of what's going to be involved. And then in the same way, collecting their signature and date stamping their consent for involvement in the whole project. But irrespective of how you actually obtain your consent, every consent must contain the following. So it must have information about the project, have detailed sections about the risks and benefits and what participation actually involved. Do you need to come into clinic? Does it require a blood sample to be collected? Does it mean linking your data? It should inform participants about the outcomes, the potential outcomes of the project, and if any results are going to be returned, whether this data or this project could lead to downstream projects, and certainly the use of data and samples beyond the scope of the original project. So following on from that, though, is the management of consent. And as I just mentioned before, sometimes projects mostly evolve over time, and new projects start that might build on the research outcomes of the primary project. And in that case, consent to continue to use a person's information or samples needs to be confirmed. And this can happen in a number of ways. Firstly, what would happen is the researchers would go back to the ethics committee with an amendment saying that we would like to extend the research aims of this particular project. And the HREC as a committee would then decide whether there was sufficient information given in the first consent form that the person has signed that doesn't actually warrant the research team to go physically back to the participants and gain a reconsent. And so the committee would make that decision on behalf of all of the participants. And that's what we call a HREC consent for further work. We also have this concept of opt-in and opt-out. It's often used in registries. So an opt-in consent is where a participant has to actively opt in or choose to be involved in a research. And the opt-out option is where you would be automatically recruited into a registry or into a project. And as the participant, you would have to actively go through the process of withdrawing from that project. One consent that has been a bit, part of the, one type of consent that's becoming really popular now is this concept of dynamic consent. And so dynamic consent is an informed consent model which enables research participants to revise and change their choices on their participation in research over time. And it's considered to positively affect both recruitment and retention of participants and the effective management of the informed consent process, particularly in biobanking and longitudinal studies where your future use of data and samples may not have been established at the time of recruitment. And that's, again, what I've talked about is the sort of these evolving research protocols. So dynamic consent has been proposed as a solution to meet both the ethical and legal requirements of the scientific research and the expectations of researchers in the use of samples and data. So I'm just going to move on now to the patient information and consent form. And I know Gethin has said to try not to use too many acronyms, but we often call this the PICF or the PIC for short. And this is an example of a consent form that I have used in a project running here at UQ for a COVID project. And essentially it's broken down into 3 parts. And so on the very left-hand side of the screen, we have the section which provides the information about the project. You know, who is running the project, what does the project involve, and what are the risks and benefits of that. The middle section of the form is actually what is the consent form, and this is where we have these statements or clauses that people would agree to and confirm their informed consent as part of being recruited to the project. And the final section is the form for withdrawal, and this is mostly the section that the dynamic consent really addresses, is managing this withdrawal from projects in a much more dynamic and real-time way. So now I want to take you through some of the key statements in a PIC, and I have identified some here in green. I'm certainly not expecting you to read those directly off the screen, but I'm going to just talk through these individually now. So the first statement, and I'm just going to read this exactly as it's written, is I freely agree to participate in this research project as described and understand that I'm free to withdraw at any time during the project without affecting my future healthcare. And so this really talks to 2 parts of those 4 pillars that I mentioned before. Certainly respecting people's autonomy to participate in projects and their autonomy to withdraw from a project without having any consequences to their healthcare or their employment, if that may be the case. The second one is I understand that all my data pertaining to me, including my DNA I've provided, but not my name or address, may be available to researchers in the future, some of whom may have commercial interests, and I donate my biological sample freely for these purposes and waive any claim to commercial rights arising from this work. And so this is about allowing researchers to share your data. It also means that we avoid duplication of work in the research space. So it's good stewardship of using public funding and avoiding that duplication. And also we're asking participants here to waive their commercial rights in the event that any of the outcomes of this research are translatable and have a commercial outcome. I give permission for my research team, doctors, hospitals and other laboratories to release information around my conditional treatment for the purposes of this project. And this again is about capacity building and making sure that the aims of the research project can really be achieved by having maximum access to all the data possible to really progress those research aims. So it's about the participants, giving their permission for data linkage. And a relatively new section of the PICF that is a directive of the NHMRC is to give participants this opportunity for a more fine-grained consent, where they could choose to consent to only this research project, and perhaps any downstream projects, and then unrelated future projects where information that's been collected in this project would certainly have use or applications to downstream projects. So gone are the days where we have one project and we have one set of researchers really trying to progress one set of aims. But to accommodate the changing research landscape, there are a number of frameworks that have been developed to best support human research and to maximise the participants' contribution in the research funding that contributes to them. So research governance is a framework for institutions to use to ensure that research is conducted responsibly and safely and is scientifically and ethically sound. And research governance also considers legal compliance, financial management, accountability and risk management associated with the research at the particular site. So the national statement has really been progressive in this space in developing ethical guidelines for a number of models. And I'm not going to go into all of the detail here really in the interest of time, but there are certainly models for registries, for biobanks, for data linkage projects, and as Colin's going to talk to you a little bit later, certainly for clinical trials as well. And I do just want to leave you with something that I feel is very important. It's all very well for us as researchers to have these national guidelines and we have the responsible code of conduct, but how do we actually implement these guidelines at the bench and when we're actually undertaking our research? And we can do this across a number of different ways, from the very first instance in terms of writing our submission for funding, ensuring that we do our budgets correctly, making sure that we allow sufficient time and funding to really embrace the new technologies and appropriate methods that we need in order to progress these aims. Certainly at the bench in all the protocols that we run, making sure that we have appropriate numbers of controls, the QC standards are high, that we have well-trained staff, and staff really understand the importance and significance of every sample that they're handling and the value of that contribution by that participant. An accurate recording of all the experiments and all of the processes that we undertake, clearly in lab books, not only for writing up manuscripts and presentations, but certainly also for auditing requirements as well. And I also just want to touch on, certainly with collaborations, most research now is multidisciplinary. It goes across institutions sort of nationally, but also internationally. It's really important for us to make sure that we use the same systems and protocols, interpretations and definitions so that any of the data that we generate is well harmonized and that we can very effectively integrate data and really progress those research outcomes that we're all aiming to do. So on that note, I'm going to finish up. I wanted to say thank you very much again for my invitation for this project, and just to leave you with some pictures of the stunning North Stradbroke Island, which is off the coast here in Brisbane. It's a great place to visit and cannot take a bad photo when you visit. So thank you very much.
Thank you, Anjali. That's an awesome overview of an incredibly important process, actually, that perhaps people don't always think, and it really undermines how much care and thought does go into design of research projects and to try and ensure that we get the best outcomes, and obviously participants in those projects are treated fairly. I mean, I think, as you said, there's been a bit of a checkered history in medical experiments previously. I had a quick question, actually, just a verification that when you sign up for tick the box to allow your samples to be used in other projects, those projects will always have equally stringent ethical approval, etc. processes to be involved. So patients can be very confident that their samples won't go off into some other less well looked after project.
Absolutely, absolutely. So even if participants agree to that data sharing or sample sharing, we actually cannot even engage in that process until all the research governances have been undertaken by that next project. They have the appropriate ethical approval for that. And we actually take the time to review the consent forms from every single individual to make sure that when we send out a set of data or a set of samples, that each person's personal preference has been accurately recorded and is included or not included in what we're sharing. So it does take up a quite a bit of time, I guess, in the laboratory or in our groups, making sure that we actively and accurately reflect people's research status and their wishes. And so, for example, we have people who would cross out statements on the consent form and say that they're happy to data share, but they only want their samples to stay in Australia, or they only want their samples to be shared and not their genetic information. So all of those permutations and combinations we accurately record in our data management systems so that we can uphold that contribution from that participant.
Great, thank you. Keith, you have a question?
Thank you. Thanks for the presentation, Angela. That was good. I was interested in the risk table where you banned the potential risk of the projects into 3 categories. The third category very high. I'm used to a breakdown in risk matrix of about 25, not 3 categories.
Yeah.
How many projects, trials with potential to do really good are in that very high category? And of those, how does anyone differentiate the stuff that's towards the bottom of that category from the stuff that's towards the top of it?
I think, so basically any clinical trial is considered high risk. Any project that has a genetic component is considered high risk. And so I think certainly in the MND space, except for perhaps where people might be filling in online questionnaires about their experiences with, you know, in care and things like that, most of MND research will actually fall into the high risk category simply because the participants who are being recruited, we would have to tick the box that these people would be under, you know, under highly dependent medical care. And so the ethics committees are very, not cotton wool is not the right word, but they're certainly put very strong fences around specific participant groups, ensuring that we have to address all of the risks and all of the potential, you know, consequences of those risks very accurately. So it certainly all the research that we do here at UQ in our MD research program, everything is high risk.
So for a dumb engineer, what's the purpose of the gradation if most projects, at least in our world, end up in that high category? The tool doesn't seem to offer assistance. It falls to personal judgment of committees and universities and things. Is that how it works?
I think it's because the tool isn't just for MND research; it's for all research. I think so. It's really you know, there's plenty of research that goes on which is lower risk. Therefore, they don't have to go through the same extent of process as this, which otherwise you put a huge burden on ethics committees around the world to approve almost every single research project that happens. So the risk is, I guess, as soon as you're up in that it's really those interventionist projects where there is that higher risk. So and as Anjali says, the research most of us are doing, it falls into that box.
Thank you.
So Colin?
For a bit over time, but I think it's important. So I'll declare a conflict. I sit on the University of Sydney HREC and I review a lot of applications. And I always have a bit of concern. And I'm interested in patient feedback here as much as possible, or carer feedback, because there's a couple of things that concern me when I see things. First of all, protocols. I love a 30-page protocol, Anjali. Some protocols run to 150 pages. So protocols can be very long sometimes that we have to read through. But there's a couple of things that worry me about consent. Firstly is the issue of consent fatigue for participants. Some consent forms, the PICFs, can run to 20 pages, depending on the clinical. Individual is expected to read through that. The second thing is there's often conflicting consents being handed to patients in a very short time, because a patient might be asked to consent to give a blood sample, consent to brain donation, consent to be in a trial. So they're actually offered multiple consent forms to review over a short time. That's a concern that I have. And then there's the issue of research overreach. So this idea, and I'm sure at UQ you're all very strict in what you ask, but we see increasingly on HREX that researchers want everything, including the kitchen sink. So they're asking for huge amounts of data and access to medical records, which may have no relevance. And now we're seeing with data breaches going on around the world, how that's all managed. So I'm kind of interested in, we don't get feedback on HREX from study participants. Anybody here who's actually has consented to research to give me their thoughts about what that looks like for them, and am I over worrying about these things?
Love to hear from anyone who has been involved in a trial and what they thought about their consent process. Was it clear and simple, or massively complex?
Guys, if I could add some flavor, I did maybe 10 to 15 research projects, and the consent for me was simple because with a science background, I understood the content. But I think if you were not so scientific, you really do need someone to walk you through the specific details of what you are consenting to and what your sample types are. But for me, it was straightforward, very logical. But like I said, it's different people with different backgrounds that is of major concern.
Thank you, Matt. Jenny, you have a comment as well?
Yes, I thoroughly concur with what Matt just said. That was my concern. I had quite a lengthy document, having just recently consented to participate in a trial. My concern was that there are a whole bunch of factors that could contribute to why somebody may not necessarily be able to either understand or necessarily be able to subsequently respond, and that could be a reason why people subsequently don't enroll. But it's just really the accessibility of that consent information. I did have one other point, but sorry, that's escaped me at the moment. Thanks.
So I guess it's almost an issue of whether that consent is truly informed or not, rather than just consent.
Absolutely.
So I might just jump back in this.
Yeah, please. Yeah, Angelique, go ahead.
So just a couple of comments. So certainly I agree about the long consent forms, and they can be incredibly daunting, and there is certainly a lot of information to take in, particularly in many cases not long after your initial diagnosis. But there certainly is movement in this space to create different types of consent forms and move away from these very long documents by having more workflow styles or other ways to engage patients or participants and talk them through their participation without having to just send them these big long documents. And so that could be interactive, there's videos or there's apps or there's, really embracing the technology and have other ways of engaging participants for projects. And that's certainly something that's coming along. And just to the other point about, a lot of projects all want, the same data, and they each want their own set. And I guess that's one of the foundations for the SALSA project in particular, because we were finding even within the Brisbane clinic, we had 7 or 8 research projects going, and everybody was administering the ALS-FRS, and everybody was asking about your diagnosis journey, and everybody was asking about your care. And so what we've done is, by combining all of those collections of data into one portal, you know, whether it be now MindAus or whether it's SALSA, that information is collected once and then it is shared many times across all of the research groups. And that really reduces the research burden on participants. But we still also then have a very accurate way of collecting and capturing that data that is going to be consistent across the study, so that we can integrate data both locally, you know, even within the one clinic, but also across the country and internationally. So there's certainly lots of there's always room for improvement, and so it is good to hear that feedback. And if anybody has any other comments, I'd be really interested to hear them, and so please do get in touch.
Great, thank you, Angeli. We'll move on just in the interest of time here. It's great to have that discussion. Now, I gave Alan the real toughie here and asked him to summarize statistics for a more lay audience. So please, Alan, take it away and take us through this potentially complex concept.
Okay, thanks, Gethin. So for people, my background is in statistics, and I now apply that to large genetic data sets. And my research is looking for biomarkers of disease so we can improve diagnosis speed and give better monitoring of disease and better ideas about progression, and also to understand the biology so we can match the right people to the right clinical trial or right treatment. So as Gethin said, I've got half an hour to teach you about statistics, which is a bit of a daunting task, but we'll go through what we're going to do. So I'm just going to move that off my screen. A lot of the stuff presented in the media, you get quite sensationalized headlines. So there was this recent one about the ex-rugby internationals having risk of neurodegenerative diseases between 2 and 15 times higher than the public's. And so this talk is about understanding the numbers behind that, so you can really try and assess the validity of the research and the strength of the associations that people are presenting. So I'm going to focus on comparing two groups of people, because a large portion of MND research is about comparing two groups. So we've got sort of an observational study where we take a group of people with MND and a group of people without it, often referred to as controls, and we compare them in some way. So we might do surveys about lifestyle questionnaires, their environmental exposures and that sort of thing, and just compare the two groups and see if we can see a difference in what they've been exposed to, and that might give us an idea about disease. So that's sort of what happened in this rugby case. For clinical trials, they're also mainly comparing two groups in the simplest form. So you'd give one group of people a treatment and another a placebo and compare the outcomes. So mainly comparing two groups. And so using statistics will allow us to compare the differences between the groups. And I'm going to cover a few key concepts of statistics, being the p-value, the effect size, bias, and replication. We'll go through each of those in turn. Okay, I'd like to highlight a bit about why we use statistics. So you do find a lot of anecdotal evidence in MND and really with any disease. So you might have heard statements like sports people are at a higher risk of MND. You know, this region of the country has a higher MND risk, or this supplement's helping my MND symptoms. And while those statements may be correct, we really want good, solid scientific evidence that they are correct, because if we've got policymakers and clinicians, they're not going to act on just anecdotal evidence, because sometimes anecdotes are wrong and acting on them can actually be harmful. So we do statistical studies to try disentangle this, the fact from fiction, or just confirm these anecdotal evidence. So we're going to talking about comparing two groups of people. And so if we just go into two groups of people, even if they're in very similar groups of people, we will see differences in there. So I'm going to go with an example. If you walk down a street and talk to 10 people and count the number of males and females, you won't get the same answer every time. So even if it was 50-50 males, females in the population, chances are you won't meet exactly 5 males and 5 females when you're walking down the street. And so this variation we call sampling variation or chance variation. So these are differences that just happen by pure chance. And statistics is really about is the differences we see between two groups more than we'd expect to see by chance. So I'm going to go through the main metric that gets reported for comparing two groups is a p-value. Now, p-values are very important in the scientific literature because they tell you what statistical evidence you have for the observation, but they're almost never reported in the news media. So what we do is we perform a statistical test. I'm not going to go into the details. I'm more about interpreting the results. And we get a value called a p-value. And for comparing two groups, we can say that p-value means what's the probability I'd see this difference between my two groups if there was really no difference between them. So going back to the walking down the street, I had, let's see if I can go back, two groups of people here that we saw when we talked to 10 people. In one group, 5 were blue, 5 were pink, and the other group, 6 were blue and 4 were pink. The p-value would tell us, is that difference we see there just by chance, or is it real? So the way p-values work is most people take a p-value of 0.05 or 5% as being something of interest. Now, that 5% says the difference we're observing between our two groups, there's a 5% chance that that difference just happened by pure chance. So if you think about that, that's 1 in 20 studies will give a p-value of 0.05 by chance. And that might not be stringent enough. And we really want to see very small p-values. And the definition of small can vary. For a particular study, 1 in 20 might be good enough. 1 in 20 chance you're wrong, and this is not worth looking at any further. For other studies, we want to be a lot more certain of our results, and we'd want a very small p-value. So 0.01, that's 1 in 100 chance of being wrong. 0.001, we're down to 1 in 1,000. So it really depends on what we're looking for. But the p-value does give you a metric of how strong the difference between the two groups is. And as I said, you'd never get this, I've never seen it in a news report, but it will be in the original publication of the result. And it's an important metric because it gives you a feeling of how good is the evidence for the headline you're reading. So the effect size is the next concept I want to go on to. So it really, now we know that the difference between the two groups is significant. We've got a very small p-value. The effect size is the next thing to look at because there's a difference between being statistically significant and being actually biologically relevant. So a small p-value only really tells us there is a difference between the groups. It's more than what we'd expect by random chance. It doesn't tell us how big that difference is. And that can be very important because, you know, big studies can give us statistical significance, but it's a very small difference that doesn't really matter in a biological sense. So there's really two ways we're going to compare two groups. One, we might look at the risk of disease in one group versus the other. So for example, the risk for the rugby example, the risk of disease in those who played rugby internationally versus those that did not. And the estimates of risk, if it's 1, that means that you've got a risk that's equal in both groups. If it's less than 1, there's a protective effect. So that says that the one group has the exposure actually protects you from developing disease. And if it's greater than 1, the exposure is increasing risk of disease. So that's if we're just comparing risk of developing disease. There's also potential ways of comparing, for example, progression rate or time to onset or many things like that, where 0 would be our baseline, where there's 0 difference between the groups. And then you can think of it being above 0 if the one group has a bigger effect than the other. So there's a wee example on the side there. This is males and females, and we can think of it as height. We know on average, males and females are at different heights, and males being slightly taller on average. And the effect size here, we can see between the dashed lines, you've got the average height for males and average height for females, and that's a measure of the effect size. Now, when we're doing research and we go out and collect 2 groups of people, find a significant result, we estimate the effect size, but we need to put it in a biological context, because as I said, really big studies can find very small effect sizes. So for example, if an exposure of some variety increased your disease risk by 1.01 times, so a very small increase of risk, that could be significant in terms of p-value, but is not really that interesting in terms of biological context, and you wouldn't see people acting on that in terms of legislation. We also have to think in context of the disease we're looking at. So if you had an environmental exposure, for example, that increased your risk of disease 2 times. Well, if it was a disease that happened at a rate of 1 in 2 million, that would only take you to 1 in a million. You mightn't be that concerned about that increase of risk. But if it was a disease that happened to 1 in 100 people, 2 times the risk would be 1 in 50 people, and that's really quite a big difference. And so the effect sizes also need to be put in context of how much of a real world difference they make, not just in terms of a number. So once we've got our effect size, we know we've just sampled from the population and we've got an estimate of the effect size, but there's actually errors to our estimate. We don't know that's exactly the effect size, because if we go out and repeat the experiment again, we'll get a slightly different result. And the way that these are presented a lot is confidence intervals. So you can think of that headline back for the rugby again. It said, playing international rugby has a risk between 2 and 15 times for a neurodegenerative disease. So that's the confidence interval. It says, we don't know exactly what the risk is, but it's somewhere between 2 and 15. So generally, we present our results as a range that we're pretty confident contains the real value. And the main way people do this is as what's called a 95% confidence interval. So it's a region we're 95% sure the actual risk is within these two extremes. So we can also look at confidence intervals and think about going back to significance. So given this example on the left, here goes a dotted line. Maybe we're comparing two groups of people and looking at the risk of disease. If our confidence interval crosses the line, then we've got an effect size that's above 1 for a risk of disease, but our confidence interval says actually it could still be below 1. So we really got no idea whether that has an effect on disease or not. And this comes back to the concept of the p-value. If you've got a significant result, it won't cross the 1 line here. So this example in the red here, we've got the effect size. This seems to reduce the risk of disease, and the confidence interval doesn't go over 1. So we're 95% sure the effect is to reduce the risk of disease here. And ideally, with bigger and bigger sample sizes, our confidence intervals get smaller and we have a better idea of what the effect size is and where it is. Another important concept in statistics is bias. So here we're comparing two groups of people, and we really want them to match in every other way except the thing we're testing. So for a clinical trial, we'll have people on the treatment and people on the placebo, and we really want those two groups to be comparable in every other way, so that any difference we do see, we can attribute to the treatment. So things like a person's age, whether they're male or female, it could be where in the country, are they in an urban environment or a rural environment? So there's a lot of things we need to think about when matching the two groups. Now, when you're seeing results published in the news, it's often very difficult to assess what you do and do not know about the groups and how well they're matched. And you'd also, they might say they're matched on age and male, female, but you don't know if anything else has not been matched on that may be important. So it's something that you can look at, but it's often difficult to interpret. And the final concept that I think is very important is replication. So for any scientific study, we really want to be able to replicate our results. So if we go out and find an association of an environmental exposure with disease, we should be able to go out and do that study, or someone else goes out and does that study in an independent sample of people and get the same result. So if a result is real, we should be able to replicate it in different samples. And that really helps us prevent conclusions being made due to a bias that we just missed completely in one population. So those are the main concepts. I know it's a lot, but there is a lot to do in stats for half an hour, but I'm going to go through the example here and see if we can apply these concepts to look at the evidence for this news article. So this was published at the start of October, and they claim playing international rugby increases your risk of neurodegenerative diseases between 2 and 15 times. So we already know the confidence interval there between 2 and 15. They've given us some information in the article, but there's actually very little details of the study there. And to really get down into the information, you're probably going to need to find the original study. There is going to be a session tomorrow, I believe, on looking at scientific media. So, but most of these studies will be available online and you can download them. If not, generally, there's an email link to one of the authors and you can just email them and say, can I have a copy? and everyone's very keen to provide them to people. So it shouldn't be too difficult to find the original study, and a lot of times the news articles link directly to them. So here goes one of their tables of the results. Now, this is a lot of information, but this is looking at the primary cause of death for former international rugby players and a matched population comparison group. So they've matched them somehow, so that's great. And we'll just look at the neurodegenerative disease. We see they had 2.7% of former rugby players got neurodegenerative disease, and 1.5%, so 18 out of 1,236 of the matched control got a neurodegenerative disease. Now they give us what is essentially a risk. So it's saying 2.4 times the risk of developing a neurodegenerative disease if you played international rugby. And they give us the confidence interval. So they say we're 95% sure this risk is between 0.92 and 6.4. And you'll see that actually covers 1 there. So we're not actually sure with this confidence interval whether it covers, whether it is increasing the risk or not. And that's reflected in this p-value here. So 0.07, so 7%. So there's 7% chance this observation is pure chance and not driven by the exposure being the rugby players. And so that's the concepts here. We estimated the effect size, got the confidence interval, and we got a p-value. That doesn't actually match the headline. So this paper actually took a bit more digging, and they considered both primary and secondary causes of death. And then they get a risk estimate of 2.6 with a confidence interval of 1.4 to 4.7. So the confidence interval is now completely above 1 and the p-value 0.02. So that gives us an idea that there isn't a statistical association with playing rugby and neurodegenerative disease. They actually had split it up into different diseases, and they've got an MND ALS, where they have the risk being 15 times, although they really have no idea because the confidence interval goes from 2 to 180 times. So that just tells me there wasn't enough data. And in fact, if you look at this cross here, they say there was 0 people that got MND in their general population sample. So that's a bit of concern. So the things I talked about as well was looking for a bias. So they did say they had a matched population group, and when you look in the details, they matched it for age. I think these were all males. And then also the region of the country they lived in. So they looked pretty matched. But of 1,236 people, no one was diagnosed with MND. And we know the lifetime risk of MND in the UK is about 1 in 300. So that's a bit weird. So maybe there's some bias going on there. And in their results, they also had this plot, which is just the risk of dying of anything for rugby players versus no one. And here again, we've got the risk of 1 being our baseline. And we actually note that rugby players have a lower rate of death. So we've got the effect size and the confidence intervals all below 1. They have a lower rate of death until they hit about 80. So the question could be, is this just rugby players living longer because they don't die of, it was mainly driven by cardiovascular disease and respiratory issues, so they don't die of that. So they've lived longer. Is that why they're getting higher rates of MND? And I think this needs to be looked at in another population. So replicating would help us understand that in more detail. And so that can be quite complicated to take a result from a newspaper or online and try to understand it. But there is very good sources online. So if you look at, so here goes a great example from the MND Australia Facebook page. They actually did a summary of this study, and saying, you know, the study does suggest there's a risk. Sample size was quite small and need more research to be undertaken for us to really understand it. And so I think it's always good to appreciate things you see on news websites. The headlines are designed to make you click and read the article. So taking a look at a less biased source of news, such as the MND Australia Facebook page or any major MND group, is a better way to get an understanding of the study. Okay, so it's a lot for half an hour, but hopefully when you're looking at results now, you can see a p-value and understand what's the probability the result they got was purely by chance. The effect size being how big an effect does it have, and the confidence interval telling us how accurately is that effect estimated. Is there bias in the study? Is there something else that could be driving the results? And looking for replication. Has this result been reproduced by other research groups? Okay, thank you.
Awesome. Thank you. Thank you, Alan. That, as you said, is a tough ask, but I think you've delivered brilliantly there. I think hopefully we have a lot more understanding. And of course, we've all been bombarded the last couple of years with medical studies and statistics about COVID and vaccines, etc. But I think hopefully the information you got there is really going to help people to put it into context and realize that yeah, you don't believe everything you read. You know, even the ABC blasts out some of this stuff sometimes, and it's not always particularly useful. Or we find that. I mean, we get, as soon as one of these studies comes out, we quite often get quite a few calls or emails, and unfortunately sometimes you have to be a bit of a damp squib, especially when it's about clinical trials or things like that. Does anyone else have any comments on that or questions, or has Alan just explained it too well and everyone now fully understands statistics? I think we might be good to, thank you, Rosalie, comment, good overview there. Since we are running a bit behind, we might roll on to the next talk now. So we were swapping the schedule around a little bit due to a scheduling conflict. So next up, we're going to have Colin Mahoney from Sydney Brain and Mind Centre, or originally from the Sydney Brain and Mind Centre, who's going to take us through the treatment pipeline and clinical trial design, which should hopefully dovetail quite nicely with the ethics and the statistics studies. So Colin, if you're good to go. Great, we can see your screen and we can hear you, I think.
Hear me, great. Okay. Let me just make this bigger. Okay, so we've had some really great talks, and actually I think everything does kind of speak to one another, and particularly coming on from the statistics. The devil is often in the detail, particularly with clinical trials. We don't overinterpret data. We've already had some discussions, you know, Richard's mentioned, you know, some problems with trials and how they're recruited and what we need to do. So we might try and flesh those things out a little bit more in the talk. So what I'm not going to do is go through every trial drug ever tested. We haven't got time to do that. Highlights of important recent discoveries, things that are set out an agenda to try and improve trials for patients, improve accessibility. And we've again talked a bit about that already and the problems there. So just to start off, I think again, because we're talking to a fairly diverse group of people, I think it's useful just to spell out what is a clinical trial, you know, and so what do we mean, what fulfills clinical trial. So it's a form of research. It involves people. So we can't do trials without people. It tests an intervention of some sort. So we have this notion that all clinical trials have to be, you know, testing a drug, but they don't. So a clinical trial can test any intervention. Firstly, and most importantly, the trials landscape is set up to establish if something is safe. That's the first criteria that has to be passed, is something safe. And then we have to decide, well, can that intervention change an outcome in some way? As I mentioned, interventions can be different things. They can be drugs, they can be surgical procedures, they can be behavioral modifications. We've heard already about and BiPAP as being important in them. Typically, we'll compare two groups, a group who gets either the placebo or what's considered the best standard of care at that given time with the intervention. And you follow those groups up and you establish after a set period what's happened to the people in one group versus the other group. Importantly, to try and avoid things like bias or jumping to conclusions, before we start the trial, we have to know what we're going to measure. So we have to set outcomes in advance. So that way things are more robust and the evidence for a particular intervention. Trials in different phases. And probably the first randomized controlled trial was probably about the 1940s, and it looked at streptomycin in tuberculosis. And really the kind of the approach to really randomized trials, clinical trials, changed much in 70 plus. That is a bit of a problem because we're probably not innovating enough to drive change and to increase because of the way trials have typically been designed. The first thing you'll see on this slide is that trials are unacceptably long for patients who have motor neuron disease. We're talking about 10 to 15 years for most trials. And so if you're picking a drug, let's say, well, then that might start in what we call a drug discovery phase, where in a laboratory compounds are being identified. Then those compounds are being moved on to test in cells or in animals to see if they have some what we call target engagement. Are they doing what we think they might do? Then we'll move on to phase 1 trials, which are typically in healthy, often young individuals and medical students who are looking to boost their income. And it was very common in my peers that people went into flu camps and things like that as a phase 1 trial. Not really absorption, safety parameters. Does it get into the system? How much of a dose do we need? We progress into increasingly larger trials, phase 2 trials. Many phase 2 trials are, the first time a patient will have contact with a clinical trial, and these tend to have smaller numbers, predominantly aimed at, again, assessing clinical cohort, also efficacy. And then again, we move through larger trials, phase 3. And then after a drug is thought to be effective and approved by a regulatory agency, we can then have phase 4 studies, which are probably becoming more and more important in the regulatory environment we're in. And these involve following participants who may be given an approved compound and trying to get that real world sense of what's happening on that particular compound. And the process, as I said, is probably too long for most individuals with MND. So what's currently approved? So what do we have? And we've heard from Richard about Relyvrio, and probably the latest thing to be approved. But in Australia and in indeed many jurisdictions, this is still the only drug approved by regulators, Riluzole. So it's been around since the 1990s. It's a modest benefit on survival, and that was how it got its approval. It was a modest benefit of a short number of months. However, it probably has consistently in different trials where we've included in arms where patients on Riluzole, it's probably been consistent in that that benefit is real. It probably is a genuine benefit about being on Riluzole, but it's still quite modest. And nowadays, it's often a prerequisite to be in a clinical trial, as it's considered the standard of care. Edaravone has been around for a while, probably over a decade, and it's approved in some jurisdictions. Again, it's not approved in Australia, and we have patients that would import it from Japan and access it, but it's quite cumbersome to administer. So in actual fact, when you talk to a lot of patients about it, they'd rather not have it because it's the way it has to be administered through a drip. And this was approved on, some people would argue, fairly weak grounds, that it looked at a fairly tight group of individuals who had milder disease, who had better lung function. And subsequently, that's how it got its FDA approval. I think very recently, in the last few months, an oral form has now come on the market, which may increase accessibility and may drive demand in other regions for patients to get edaravone. And then as we've heard from Richard, Relivrio has just been approved. It's the latest drug, just September 2022. And this is a combination of two existing drug compounds, sodium phenylbutyrate and taurursodol. I can never say it properly. These are two existing drugs. Both have been around for decades, and the drug company involved, Amylyx, put them together in a trial. And as we heard, there was some benefits in the phase 2 trial of a reduction in the rate of decline on the ALS-FRS, which led the FDA to approve it. But it was a close decision at the FDA. I think it was a 6-4. It wasn't a unanimous approval. So we've seen drugs now getting approved on phase 2 data, whereas previously regulators often wanted much more longer-term data. These drugs, with the exception of Riduzol, are all very expensive. And again, as we heard, insurers in the US are looking for reasons not to fund $158,000 a year drug, particularly when the drugs have been around for a long time. You have to question the ethics of those figures. However, the drug companies would argue that the cost of running a clinical trial is exorbitant. Really, to be sustainable with a rare disease, they are the types of figures that you're looking at in terms of charging. So as a reminder to everybody here in Australia, listening in Australia, only Riluzol is TGA approved. I've had patients who've gone out and bought various concoctions to try and make up drugs. You should always talk to your treating teams before doing anything like that, because there can be unforeseen side effects. But really, we're really lagging behind other jurisdictions in getting drugs which have evidence bases approved. One of the problems with drug development and clinical trials in ALS is, in order to have a drug may work, you have to have a really good understanding of why a person might get a particular disease. And that's been a challenge in ALS. And there's different pathophysiologies suggested, but fundamentally, we still don't have a very accurate and robust concept of why people get motor neuron disease, at least the 90% of sporadic cases. And it's probably a multifactorial disease. We heard about risk factors from environmental exposures like head traumas. That's been very topical lately. We know that for a long time, changes in how calcium moves around motor neurons, leading to too much of this chemical glutamate, causes motor neuron death. More recently, we've started to look at other pathways, particularly autoinflammatory processes and upregulation of certain cell messengers, and whether blocking and targeting these cell messengers and reducing inflammatory responses may have a role in reducing cell death. And we also remember that motor neuron disease was probably, the shift has focused more on the brain and the spinal cord. There is still a component which is a lower motor nerve or peripheral nerve problem, and there's been some evidence to support increasing muscle contractility through troponin binders to try and improve the force of contraction and in doing so improve things like breathing and strength. So this challenging landscape, which does make it hard to figure out what drugs are going to work, what drugs do we choose next for a trial, and there's been no lack of effort really in terms of the number of trials that have been done, and particularly in the last few years we've seen an acceleration in clinical trials, and various trials have looked at various pathways, whether it's glutamate, whether it's inflammation, whether it's neuroprotection, and you'll see here that the bulk of the trials have unfortunately failed. They've not met that outcome that was set when the trials started. There's been a mixture of how these trials have been funded. So trials can be funded through industry. So a drug company can come in and say, we've got a compound, we want to test it out, and then go to clinicians and say, can you help us recruit patients? Or they can be academic, and academic studies tend to be smaller. They tend to be well resourced, but they're also important because often academic studies are kind of starting point for larger studies, because there's increased flexibility of how a clinician might want to test a particular intervention. But broadly, and unfortunately, most of the trials that I've shown here have been negative. So this slide is a few months old, but at that time there were around, and this is from clinicaltrials.gov, there were around 300 trials globally that were registered on clinical trials. Met that outcome that was set at the beginning, the primary outcome. So obviously we've talked about riluzole being one, edaravone, masitinib met its endpoint, and more recently then Relyvrio has met its endpoint, its primary endpoint. But you can see that the problem is there's a lot of failures. Outside of ALS and other conditions, there's been similar levels of failure. So in my other hat as a cognitive neurologist, and particularly in Alzheimer's disease, we've had similar levels of disappointment. So neurodegeneration has been a hard nut to crack. That may be relevant to an Australian audience, because many sites around Australia were recruiting for these studies. So ReFALS a few years ago, and this was looking at a drug to improve contraction in the muscle, to see if that it improved breathing. For those affected by SOD1 mutations, we've had the antisense oligonucleotide tofersen study. Again, it didn't meet its primary endpoint, but the company themselves are suggesting that in some individuals, particularly those faster progressors, which they really tried to heavily recruit, that they may benefit from this medication. And again, it goes back to the point that Terry mentioned earlier, which was that the trials are just too short in some instances. So some individuals with SOD1 mutations can live a really long time, and you need a long period of observation and engagement to measure outcomes in those individuals. Another trial I was involved in, ravulizumab, which is worked on a thing called complement, which is a part of the immune system. Wasn't showing any efficacy, which was disappointed. And some people will be familiar with the NurOwn as well, which are patient-derived stem cells. Again, some signals of early efficacy, but it didn't meet its primary endpoint. So there's some more recent trials that haven't shown efficacy. The Healy platform, which I'll mention in a moment, which many people will be familiar with, was a really new and revolutionary way of trying to test drug therapies. They've already cycled through some drugs, so they had applications at the beginning from different companies to kind of submit a drug into the trial, and two of the drugs have kind of met having them for 6 months. The data has been analyzed, and these two drugs verdiperstat and zilucoplan both failed to meet their endpoint. So they've been dropped from the study, but the idea being you drop them out and bring new drugs in. We'll yet to see if that's the best way of running a trial, because again, that goes back to the point of, is it too short a period? But the argument being that it's a quicker way to test more therapies. The project that I was involved with in Sydney was called Rescue ALS. That trial is essentially completed now. There is still some patients in an open label extension. This was a trial looking at something called CNM-AU8, which is a nanocrystalline form of gold. The idea being in models and animal models of motor neuron disease that this boosted cellular energetics or function within the motor neuron and reduced cellular stress by removing some toxic species and trying to keep the motor neuron alive and functional. So it was a fairly small trial with about 45 patients recruited to look at something that hadn't really been looked at before, mainly to power the trial, because it was a small number of individuals, and looking at clinical outcomes like decline on the ALSFRS requires bigger numbers. So this trial was designed to look at something called the motor unit number estimation, which is a nerve test which counts the number of motor nerves supplying a group of muscles. And we thought that might be a more sensitive or early way of assessing a change. So that was set up as the primary outcome measure. And then the secondary outcome measures, we use the ALSFRS and other quality of life measures as well. And unfortunately, it didn't meet its primary outcome measure. So we didn't see a change in this motor unit count that reached significance. Though when we looked at patients who were had limb onset, so problems starting in their arms or their legs, and looked at patients who had bulbar onset, which starts in their swallowing muscles, there probably was just about a difference. And so one of the problems with using motor unit number estimation was that we relied on looking at the arms and the legs. It's quite difficult to study the muscles around the mouth. So that was a flaw in the design. However, some of the secondary endpoints were more promising. And what we saw was that over the 36-week period, that there began to be, over time, so again, this idea of following people a bit longer, we started to see a separation. And the idea being here, what we call event-free episodes. So that's not requiring a PEG, not requiring BiPAP, and not dying. And we started to see a difference at around the 24 to 36 week. And then when patients who'd been on placebo from the beginning and then transitioned to the open label extension where everybody got the drug, we found that there was a difference. If you were on the active ingredient from the beginning all the way through, there did appear to be a survival benefit of about 65, 62, 63% if you had been on the active drug all the way through. So major cautions, it's a small sample size. We were overrepresented with bulbar patients compared to how often bulbar patients actually occur. So we had some overrepresentation of some groups. However, it's somewhat encouraging. The Healy platform are using the same compound, and they've actually shown fairly similar findings. They didn't meet their primary endpoint, but they did see that patients, again, on the active treatment were less likely to die or transition to permanent ventilation. So they're continuing. So they haven't dropped that yet from their protocol. They're continuing on with that study. So I think it's a therapy. Pivoting away from drugs for a moment, well, not necessarily drugs, but pivoting away from disease modifying agents. I think there's one area that trials has not had much, at least trials in ALS, has probably been a little bit neglected, is looking at overall symptom management and quality of life. So most of the trials are geared towards progression on these measures like the ALS-FRS or survival, but we tend to put less importance on quality of life or symptom burden. And in our breakout session earlier, we were talking a little about, research on carers and the role carers have in helping and supporting people with ALS. And actually, if we look more closely at tools and aids to facilitate better care in the home, better quality of life, would they make a difference on ultimately on survival? And I think it's a neglected area, and it probably is an opportunity for us all to maybe think about how we can bring trials that look at those aspects. And remember, most of the benefits from trials that we've seen have been small. They've been a matter of a few months. So we haven't had a blockbuster drug. We haven't had a breakthrough drug, which has really changed the life expectancy significantly of patients. But if we come at it from different angles, then we might actually cumulatively make a difference. So one interesting trial that's underway, I believe the team at RPA are recruiting now for the Switch 2 study, which is looking at implanting an electrode, what's called a stent trode, over the motor area of the brain to allow individuals to basically have a computer-brain interface to communicate or carry out activities or tasks where they have lost function in their arms or maybe lost speech. So I think there's a lot that can be done outside of the area of just modifying the ALS-FRS and actually looking at things that improve quality of life, improve symptoms, and I think more needs to be done on that. Currently recruiting trials. These are more relevant to Australian audiences, but we have the Lighthouse study, which is a study looking at Triumec, which is a drug that has been used for a long time in HIV. And this was born off the back of a smaller study, a phase 2 study, which didn't have enough participants. It was a smaller study, but what that study showed was that a protein in the urine that's important, that seems to be important in ALS, called urinary P75, was altered in those individuals who got the active drug Triumec. So this trial is now recruiting individuals in a larger study. Does that translate into actually clinical benefit? I mentioned a previous trial called REFALS, and this Courage ALS is using a similar drug to see if we can improve muscle contractility. Though the phase 2 trial again was negative, what drug companies often do is when the trial is negative at phase 2, they'll often go back and kind of really go through the statistics and drill down and try and find some signals in the data that might support the drug being continued. Now you can argue the merits of that because there's in some cases there's commercial interest. A lot of money has already been put into a drug and into a pipeline, and so it's in the company's interest to try and continue that. So you can argue the benefits of looking back on things and then finding things that you weren't previously looking for. But anyway, they've done that's essentially what's happened with the Courage ALS trial, and that's recruiting at the moment. Some more interesting trials and pharmaceuticals have before that Ionis have looked at these things called antisense oligonucleotides. So for people with genetic forms or familial forms of ALS, trying to reprogram the genes to work normally. And so we've had these studies for a while in Huntington's disease. Unfortunately, we thought there were going to be blockbusters in Huntington's. That hasn't been the case. But again, with everything, when they start out early, tweaks are made, dose finding strategies are revised, when to give these treatments are also revised. So I think they're theoretically excellent molecules and what they're set out to do. So we'll see with C9orf72 therapies now started. We'll see what those trials bring. And then there's a lot of other trials, and a lot of these other trials are so we heard a bit about cannabinoids earlier. Metflex, I think, is a University of Queensland study, and it's going to look at reducing hypermetabolism and see if that promotes improved clinical outcomes. So there's a whole heap of trials that are either recruiting, about to recruit, and the message really is that there's a lot happening in the space. So for those living with ALS, it is a good time to be involved in trials, because there's options, there's hopefully broader inclusion criteria across different trials that allow as many people as possible to participate. I mentioned AMX 035 earlier, and that there is going to be a phase 3 trial of that, and I think that may have been a requirement of approval. So there's an ongoing trial to look to see if those benefits that we're seeing in the phase 2 are translated. Oral edaravone has been approved in the FDA, but there are still some trials set to run to satisfy regulators in other jurisdictions, particularly in Europe. And I believe AV Science are talking about doing a mazetinib phase 3 trial. I mentioned this is a drug that was shown some efficacy in a previous trial, but they're now looking to develop a pivotal those results to try and seek regulatory approval. In clinical trials. So one of the things that we talked a little bit about is outcomes and how do we measure success. And regulators want studies to have predefined primary endpoints. They want to know before a trial starts what we're looking to see at the end, and that's reasonable. Otherwise, we go back and start changing things. But there's been a lot of debate about, well, what is a meaningful endpoint and who defines these endpoints. Traditionally, it's been clinicians, academics, industry, and patients have had less of a role in deciding what is clinically meaningful for them, and that's probably something we need to change. Also started to move more towards that might predict future problems. So trials, again, have tended to look towards the end stage of a disease. In again, most neurodegenerative illnesses, that if we wait too long to start a treatment, so if the horse has bolted, if you've lost 50% of your motor neuron pool, then maybe it doesn't matter what you give because it's too late. The process has commenced and passed a critical threshold. To try and use surrogate markers, something either in your blood, on a brain scan, something about your genetics that will tell us something about what will happen to you in the future, so that a therapy can be given as early as possible. And again, speaking to Alzheimer's disease, the recent approval last year of Aduhelm, which is a therapy for Alzheimer's disease, really relied heavily on surrogate biomarkers. It didn't rely on cognitive change. It relied on the presence of amyloid on a brain scan. So whether we can develop markers in ALS to allow us to treat individuals earlier, and then that might improve outcomes. As I mentioned, we don't really understand why people get ALS. So to choose the right outcome, you have to have that knowledge and use that theoretical knowledge to then determine what's the best outcome. So because there's a lot of basic science work ongoing, I think it's an area that we constantly need to refresh and update to see if we are using things that are fit for purpose. And we heard a lot about ethics earlier, and I think ethics are increasingly important, because what's important to a clinician or a pharmaceutical company may not be important to a patient. Equally, some of the outcome measures are pretty catastrophic. Time to tracheostomy, time to death. These are pretty awful outcome measures. And is that really, the best we can do? Again, bringing patients into the design process, I think, will help us do better in that regard. In terms of the outcome measures that we use, you can see this is an example of outcome measures. It's probably not a complete example, but amongst trialists, we don't even know what the best outcome measure is. And all of these outcome measures have strengths and weaknesses. And it may be that we need to use more complex combinations of outcome measures to try and get to the core of whether a drug is working or not. And I think people heard about the ALS FRS being a bit noisy, and this is an example of this. And I think this is North American data, which shows how noisy the ALS FRS is. And this is typically used as the primary outcome measure in most trials. So you could probably argue, just looking back at it, yes, there is a trend. And the trend seems to be, it gets smaller with time, about a point drop per month on average. But there's a huge amount of variability. So you can see some people's scores actually went up. So these outcome measures are very, very crude and very, very noisy. This is of course that we're now, as we've heard with previous drug approvals, companies want to select a specific cohort of individuals who are likely to respond to a therapy. And there's nothing wrong with. So in cancer, molecular genomics has really revolutionized cancer therapies by identifying individuals with a particular genotype that are more likely to survive if they're given a particular chemotherapy. So in populations is a way of boosting the chances of success. But that comes with a downside, because that means that some individuals won't be eligible for a particular treatment. So we have to make sure enrichment strategies don't cause some individuals to lose hope and be forgotten about, essentially, through trial design. Enrichment strategies might use different things. And again, this comes back to the idea of understanding the reasons why we get ALS, so that we can develop better algorithms and biomarkers to predict who is going to progress quickly, who's going to progress slowly, who's going to respond to treatment A, who might respond better to combinations of treatments A, B, and C. And the reason we need to do this, again, at the very start of today's session, we heard from Richard about the very poor uptake of patients within the global ALS community who are in clinical trials. So I did a literature search a while ago, and just a kind of back of the envelope calculation suggested that only 2.5% of patients globally with ALS were in a trial. So we have to ask ourselves, why is that? And how can we improve that? And if we get higher numbers, we're likely to drive better statistics. So and reduce the noise. So that benefits us all. It benefits patients, it benefits pharmaceutical companies, it benefits clinicians. It's also typically been overrepresented by some and underrepresented by others. So a big problem is that there's probably too many white educated people in trials, too few and linguistically diverse populations in trials. So that is a problem, because we don't know what's happening in certain phenotypes. Inclusion criteria. Why can't we just let everybody into a trial who wants to be in a trial? Well, mentioned earlier, because predominantly safety concerns, and also trying to test, if the drug works better in one group than another. But excessively harsh exclusion criteria creates unnecessary barriers. And again, we always, when designing a trial, we want to try and make it as open as possible. And one of the rationales for developing a new clinical criteria for ALS, the Gold Coast criteria, was to try and do that, to try and reduce the thresholds that you had to meet for being diagnosed with ALS, so that people earlier in the disease state could be included in trials. Again, as we heard from Richard, and repeating a lot of the points he made, is that having very restricted inclusion and exclusion criteria can affect the generalizability of a drug. So within bigger populations, we don't know what's actually happening. And again, there's been a move in Alzheimer's to try and develop databases now, given that Aduhelm has been approved, to actually try and develop databases and sharing strategies so that people who are on these drugs can be followed and can see what happens in the general population. And so that when a drug is approved, that there is a need for ongoing surveillance, real world data, phase 4 type follow-up. So just a few last points. I don't know how I'm doing for time, but Gethin, please tell me if I'm going too long. We are moving more towards a precision medicine approach for trials, and that's probably a good thing. We're trying to get more of patients and then give them treatments which are more likely to work for their particular pathophysiology, recognizing that patients are different, genes are different, changes in the brain may be different from patient to patient. So we are starting to move more towards this precision-based approach. And as I mentioned a few times, the Healy platform was a way of trying to combine more rapid throughput of clinical trials, but even adding to that, trying to add personalized medicine. And this is something that in Australia, we're going to be involved with lithium to try and identify individuals who carry a specific risk gene for ALS who seems to respond to lithium. So using these trials to identify people with a similar makeup, giving them a treatment which we think will work better for them, but then getting rid of that treatment pretty quickly if it's working, and then adding in a new treatment. So this whole idea of both combining precision and form type trials to really try and move that process from 15 years to 3 or 4 years. So I mentioned the Healy platform, the Magnet trial, which will look at lithium and individuals with UNC13A mutations, and I think SALSA have been very important in identifying and cohorting these individuals. And in the UK, the MND Smart platform trials, which are going to use two existing drugs, memantine and trazodone. So we are going to cycle through therapies a little bit more quickly. I think areas exist, and this is again areas where I think really patient advocacy are probably going to be critical, particularly in Australia. All the evidence, you can pass through different phases of trials, but getting a drug approved and funded is a whole other ball game. So regulators quite rightly set high bars for approving a therapy, but when you're faced with a life-limiting illness, it's important to reflect on whether those bars are appropriate. These have different rules, and that means, particularly in countries like Australia, we're often the last to kind of get a look in for a particular drug because the market share here is so small that drug companies kind of, really have to be persuaded to submit applications to the TGA. And those national variations create problems. So how can the TGA for rare diseases be pushed towards, accepting recommendations from elsewhere? And I guess that goes from the small versus large markets. I think we also have to be careful. Whilst advocacy is very important, it shouldn't replace hard data. And again, using Aduhelm as an example, there was significant lobbying undertaken by the Alzheimer's Association and patient groups to have Aduhelm approved, whilst most of the FDA panel were initially quite hesitant to do that. The recommendations they got were not very encouraging, and even talking to colleagues, most colleagues don't think that was the right thing to do. So we have to get the balance between advocacy and the data correct. We can't be swayed too much the other way either. And as I mentioned already, we need ongoing large studies, particularly with real world observations. So I'll probably, my thing keeps getting stuck, so I'll probably leave it there because I wanted to, one other thing I wanted to mention, beg your pardon, was boost clinical trials. And I suppose I had two ideas here. One is, do we go down more of a low-tech approach, or do we go more of a high-tech approach? So clinical trials are really expensive to run. They take a long time. They take a lot of governance and regulation. And we've heard already about things like ALS Untangled, which is trying to really bring data to patients and carers based on those real world experiences and perhaps smaller trials. But I think what we should be trying to do, at least at a low tech level, is trying to look at drugs which we perhaps think are safe, maybe exist for different reasons, and try and develop fairly simple and open sourced protocols and trials with centralized ethics and governance and fairly simple outcome metrics to try and get as large as possible a cohort of individuals in to then look at those benefits. So a more simple, pared back design, but using safe or I think COVID has really kind of spurred us along to develop different ways of looking at patient outcomes, what we call patient-reported outcome measures, and how we can use IT, iPads, home monitoring, creative ways of monitoring patients to try and make it as easy as possible for people to take part in trials. So that might be a good way of running a trial for perhaps people with more established disease. Then you approach of really trying to find the individual patient to test out new and innovative therapies on that group of individuals. In those situations, you probably do need greater governance and safety protocols in place, and that's where those kind of high costs are going to come in, and perhaps having a smaller sample size to kind of make trials sustainable and feasible. But anyway, they're just two ideas for kind of discussion. But overall, I think the landscape is very promising. There's a lot of trials going on. There's a lot of data out there. So hopefully we will only have more and more drugs and therapies approved in the coming years.
Awesome. Thank you, Colin. That's a fantastic overview of the treatment and drug trial situation. There's, as you say, I think it's been the best it's been for a long time. There's a lot of drugs out there, and perhaps the drug regulation authorities are starting to change their attitudes. And hopefully it won't go too far the other way where we start approving ineffective drugs either. But I suspect there's probably a few thoughts around that. Do we have any burning questions before our final presentation of the day?
Colin, I've got a question for you. So I mentioned in the beginning, that we have this peace committee where we have different stakeholders, mostly from North America, get in front of our research ambassadors once a month. I mean, certainly we'd be open to having folks from other countries come and present, but do you think it might be a nice thing to have in addition, like an Australian peace committee where you and some of the other prominent researchers from the country get together with your new research ambassadors on a regular basis to talk about projects in development, get their feedback, that kind of thing?
Yeah, I think absolutely. I think we need to have, we're a small country, so that actually should benefit. We're a large geographical country, but a small population, I suppose. And most people know each other. So that's already a kind of benefit. It's a tight community. Most people work quite closely. There is the other issue of being in a smaller environment. People want their bit of turf and their bit of territory and their bit of prestige in running something. So we have to also break down barriers between different groups and make sure that we're all in it to really help patients and their families. So I think absolutely bring people together, thrashing these things out, but also having a kind of a manifesto almost that you can go and say, we need to go a bit like I think what the Healy people did with going to the FDA as a group. Maybe you were involved in that, Richard, but going to TGA here in Australia and actually having a meeting with them, bringing people together, bringing advocacy. I think it would be a very empowering thing.
Thank you. Peter, you had a question as well, or comment?
Yeah, unrelated to my life, what's everyone's thoughts about getting genetic testing? Is it a worthwhile process to be done, or does it make any difference?
I think genetic testing does make a difference, and I think that is a personal view. We've heard a lot already about individuals who a lot of family histories in this group, and there's significant family histories. It's still worth pointing out that the majority of people don't have a very strong family history of motor neuron disease. But really, what I think we need to do in neurodegeneration is get there as early as possible with an intervention. And so if you are in that group of individuals who has a familial form of ALS, and the trials are moving to try and treat people before they develop symptoms. And what we know in other areas, that seems to make a difference. So the earlier you treat, it probably does make a difference. So having that information is probably important. But I think, again, I see it in clinic, a lot of patients, some people, I don't know what Richard and Terry their experience in North America might be, but it seems to be some patients are very, yes, I want to know, and absolutely, please tell. And some patients are, no, do not want to know. And it seems to be like light and day.
Yeah, I had no genetic. My family has nothing. I'm the unlucky one, I guess. But yeah, no history at all.
Peter, I'll just weigh in. I mean, I don't practice a paternalistic style of medicine in my clinic. I try to present options to my patients, and I try to make sure they understand what the option can do for them. So as far as genetic testing, I mean, there's several benefits that you can get from that. One, it's the only test we can currently do that might tell you why you got ALS. And that is a burning question on the minds of a lot of my patients. It certainly would be for me. Two, families often want to know what is their risk, especially young families, where there might be some family planning that's still going on.
Yeah.
That's the only test I can do there that helps with that. And then I think three, most importantly, we're coming into an era now, certainly an era where there are trials, unique trials that can be offered to people with certain mutations. And maybe even more exciting, as you just heard, if we identify a family that's got a mutation that didn't know about that, now we empower family members with no symptoms to potentially participate in a trial where they might never get ALS because of what they take. So, I mean, to me, it's a hugely important topic. And, the main downside of genetic testing used to be the cost, but at least here, there's multiple companies now that will do panels of 30 or so genes for free. So there's really no cost to most patients anymore.
So that takes us really well into the next talk. The last talk of the day is on genetics of MND ALS. So sorry we're running a little bit late, but I think it's because we've had some great discussion. We've got Marcel and Sam from GenieUs and UNSW to take us through. So whoever's going first, I'm not sure. Share your screen and if you need to, and off you go. Thank you. That's working fine, your share, Marcel.
Great, and can you hear me as well?
Yeah, we can hear you too. All good.
Oh, brilliant, winning. Okay. So thanks, Gethin, and thanks everyone for the opportunity to give a talk about this today. So Sam and I, and we'll switch over partway through. We're doing a bit of a tag team. Hopefully, keep things interesting through this talk. Appreciate it's the last one of the day. So GenieUs is on a path, a very ambitious path to seek to both identify new genetic biomarkers in ALS, and as well as use those to guide and develop new treatments for disease for ALS, really taking, I guess, the paradigms of precision medicine into this phenomenally challenging disease. We've seen a lot of success in the application of genetic and genomic testing in the cancer medicine space, but I think needless to say, the application of that in ALS is still a long way behind. But I think from what we've seen in cancer, there is a lot of promise. But it will be perhaps not a linear path, and there's a lot to be learned on the way. So I'm just going to do the introduction to this, the background and some of the genetics, and then we'll switch over to Sam to go into some of the detailed work that that GenieUs has been doing in this space to date. So very briefly, and this is just to really set up what genetics is and how we can start to apply it. So this sort of traditional, I guess, dogma of molecular biology is the DNA, which is the genome, which is effectively encodes so-called RNA, which is another type of nucleic acid, another type of molecule that we have in cells, and that in turn makes protein. And if we look at this is a common analogy that's sometimes used, where we can think of DNA as kind of like being the planning document of a living system, that the RNA perhaps is like the blueprint that's kind of read out from the appropriate chapter, whatever it might be, of this textbook or urban planning book, and then we build something from it. And in essence, what happens, and it's of course infinitely more complicated than that, but that's putting it into a very sort of, I guess, a very simplistic framework, is that the DNA, of course, encodes many different things, and the ways that it can be affected and mutated are many and varied. The single base pairs of these 3 billion base pairs of information can change. We can get things getting swapped around. We get language itself being modified and changed. It gets translated then into RNA, as we said, into this next type of molecule, and it itself is again can be exposed to all sorts of changes. And finally, the protein can be exposed to changes as well. So each of these elements are areas where things can go wrong and can be the I guess the basis or the manifestation of disease can arise from. And so when we're looking at ALS, we really need to look at more than just the DNA. We need to look at the DNA, the RNA, the protein, how this whole system sits together to start to be able to unravel this very complex picture. So to extend the metaphor, with urban planning, you can see you get a typo in there. We've turned it now to urban planning, and it's you know, the blueprint now looks a bit wrong, and then the protein comes out wrong, or it might turn up, you know, some of the part is turned around, spelt backwards. We end up with things that are the wrong way around, and everything can go in, or you can get up with the wrong numbers of copies of things as well. So there's all these different ways that errors in DNA can manifest into downstream problems at the end in cells that lead to disease. Of course, sometimes this can be catastrophic. So with that sort of, I guess, a brief introduction, I guess just to say, what is a biomarker? When we talk about a biomarker, there are two types: diagnostic biomarkers and prognostic biomarkers. A diagnostic biomarker is one that is just a naturally occurring molecule. It can just be anything really that's existent in a cell. It could be a gene, or it could be a particular protein that can be detected. And the important part about a diagnostic biomarker is that it's something that that positively correlates with a pathological process or disease. So that's kind of when we're thinking about a diagnostic, and a prognostic is something that can predict what might happen. So that might predict that the onset of disease, or it might predict what the pathway is for disease. In the cases of something like ALS, which is this incredibly diverse disease that can take many different paths, and remains a huge unanswered question, especially for in the clinical space, of when you get a diagnosis of ALS, what does that actually mean for you? What does that mean for your family? What does that mean for the progression of your disease? And at the moment, there's very little knowledge about that. So both of these areas are really underserved, and there's, I think, a lot more that can and needs to be done to benefit this. So briefly, in this perhaps addresses the previous question, is that there are a substantial number of genes that have been identified that are biomarkers of ALS. And what that means is that if there is a particular mutation in one of those genes, that if you carry that mutation, then you'll have an extremely high chance, or in some cases even a guaranteed chance of likelihood of getting ALS as a result of that. So as you can see, there's quite a large number of genes known. Now, having said that, that only explains a relatively small proportion of cases of of ALS. So in fact, only about 15 or so %, I believe, of ALS cases are ones where we actually understand what the genetic basis for it is. That is, if we sequence the genome, we go, okay, it was this gene. It's the error or the variation in this gene that's caused the disease. So some of the shortcomings that are there at the moment is that there are, as I said, there are no useful biomarkers to tell us about what the onset is of ALS. So that as you might carry a gene that will predict that you get ALS, it won't tell you when. We know very little about how what those variants will mean in terms of what the rate is or the progression of the disease will look like, and again, very little in terms of what we can do in terms of which which therapies may respond better for certain versions or variations of the disease than others. So these are all the things that I think there is a lot of potential where genetics can really help for this, because there are dozens, potentially hundreds of different genes that can cause ALS. We actually believe is that it's a very diverse disease, and that as a result of that, it's unlikely that there will be a single treatment that cures ALS for everybody. And rather, what will happen is that there will be different types of treatments and different types of therapies that will target the particular variety or subtype of ALS, which is very similar to what we're seeing now in cancer. You know, the drug to treat or therapy for a breast cancer will be very different to a prostate, very different to a lung, very different to stomach, different to bowel. They've all got different treatment regimes associated with them. And now many of these are being guided through molecular information. So, as I mentioned before, there's a big missing heritability that's still not well understood in ALS, where only 10 to 15% or so of patients have these so-called fully penetrant causative variants. That is where you have the variant, you're guaranteed to get the disease. And we know that there's that the genetic contribution still remains considerable as we see it running through families. So just the last bit that I'll mention here is just in this pathway towards these of what's happened to date. So there's been extensive so-called GenieUs studies that have been done, genome-wide association studies that you might be familiar with, which has given an indication or an insight into different parts of the genome that are associated with ALS. Now, this has told us that that it is an hereditary phenomena in many conditions, for many individuals. But what it doesn't tell us is exactly which gene or which part of the genome is actually causative. And this is sort of where the next step is now, and I guess this is approaching more the state of the art, is where we sequence individual families, so-called pedigrees, of where there are multiple affected individuals with ALS, and then by sequencing the different individuals that are affected or unaffected, we can then track down which individual what gene or what particular variant might be driving the disease. So that's how we can then achieve a very accurate diagnosis. Potentially from there, and from that, be able to link that back to a therapy that will hopefully have a better chance of treating the disease. So with that, I'm going to now switch across to Sam. Sam, if you can switch on your mic, and he's going to pick it up from here and start going through some individual case studies that have been identified by GenieUs and showing where we're at so far with this. Thanks, Sam.
Awesome. Thanks, Marcel. G'day, everybody. I'm just going to quickly pre-apologize for the coughing that's inevitably going to happen. I'm just getting over a bit of a cold at the moment. Yeah, so what Marcel's taking you through is really sort of a grand strategy stuff that does guide what we do. And I'm a bit more like the boots on the ground. So we'll get into a bit more detail about what we're doing to address these shortfalls that we're aware of. So what we're really going for is a mechanistic understanding now, something where we know what molecules are causing the problems for the motor neurons, for the other cell types that share the brain and spinal cord. So the first and one of the most important things that we want to know more about is clinical modifiers. So these are genetic variants that don't necessarily cause ALS, but they do make it worse in people that have been diagnosed with that disease. So if we just go back one, Marcel. So they're useful, obviously, because they deepen our understanding of the disease. They can help us guide therapeutic decisions, and they may become therapeutic targets themselves one day, with the best example of this kind of pathway, if you like, being UNC13A, I think. So this gene has lots of different variants in it. Some of them are really, really common just among all kinds of people everywhere. But there's been two of these quite common variants that were associated with shorter survival time in people who are diagnosed with ALS. But the really interesting thing was that ALS patients with these common variants actually were able to respond positively to lithium carbonate treatment, where other ALS patients just couldn't derive any benefit from it. There, next slide, thanks, Marcel. So another thing that we like to consider is polygenic disease. So what we've talked about mostly in the previous sections is these highly penetrant variants that give a near complete or actually complete chance of developing ALS if these variants are present. But as Marcel mentioned, that doesn't describe a lot of ALS patients at the genetic level. So we're looking more into this idea of polygenic inheritance, where we have, you can see here, a couple of different variants, like from the green down to the light blue in the polygenic column, that all contribute varying percentages of risk for developing ALS. And of course, when they're all present, that risk is 100%. Next slide, please. So taking this polygenic disease idea and the concept of clinical modifiers both into account, we're developing a perspective that looks a bit more like this, a more integrated understanding, if you like. So this is where we're assessing multiple genetic features that might be necessary to understand the disease in individual patients, or perhaps small groups or clinical subtypes of patients. So it's going to let us identify these multiple genetic drivers of disease and characterize them. And you can see on this little graph here, we've shown like genetic variant 1, full penetrance, as it was in the previous graph. But we're looking at other features like, that the type of motor decline, whether or not there's behavioral symptoms present, and the overall rate of progression are just a few characteristics that we should probably think about. Okay. Next slide, please. So so to develop this more complete perspective, I'm going to bring in now the idea of the multi-omic perspective. So what we've talked about mostly has encompassed single nucleotide variants or repeat expansions, when we've been talking about those really high penetrance and arguably causative genetic variants. So we're really dealing now with the question of disease coverage, right? So almost all of the genetic variants that we understand in ALS are these SNVs or repeat expansions. There's lots of different types of genetic variant possible there. You can see there's another 3 there that we haven't really talked about yet. And they may very well have a role to play in ALS. Now, despite there having been lots of GWAS studies and family pedigree studies completed already, we still haven't identified that 60% missing heritability that we all know we're chasing in the field. Next slide, please. So knowing that any of these types of variant can influence ALS development and progression, that brings us to the question now of personalized medicine. So with the difficulty in finding these ALS causing variants, even in really large population studies, it tells us they're just fundamentally pretty hard to discover and they might not be very common. So we think this means we have to take a personalized approach, or at least a somewhat personalized approach, to discover disease-driving genetic variants in ALS patients. So this will help us work out the disease mechanism. And once we understand those mechanisms properly, we can come up with actual therapeutic strategies that are a bit better targeted than they are at the moment. Next slide, please. And the last one on developing this multi-omic perspective is that these variants, we've seen them all sort of in this sort of trident shape at the moment, forking out from their individual levels of biology, DNA, RNA, and protein. But the fact is, variants at these different levels actually influence each other, just to make this even more hideously complex than you might have thought from looking at the first part. And they're disease relevant. We know that some of these things are disease relevant, and we know that there's more to find. So to understand these consequences, we look at these flow-on effects of genetic variants, and it would be a really big help for us to understand these to describe the disease mechanism in individual patients, or perhaps in small subtypes, clinical subtypes of patients. With that knowledge, that's how we're going to propose those new therapies. And this platform that we've been developing, we call it DIGAP around our office. It stands for Deep Insight Genomic Analysis Platform. This is how we're trying to fill those gaps and develop that perspective that we think we need. Next slide, please. So this is what it looks like. In the inputs, you can see that we've got all of those different types of genetic variant that were on the previous diagram. They run through our core process. And I won't go into details about that. It is, we would be here for the rest of the day if we're doing that. But the headline is it gives us these outputs, like genetic variant prioritization is probably the biggest and most useful one to us at the moment. And we're expanding to look at this genetic variant burden as well. So the prioritization is about finding sort of the likely highest impact variants in individual patients and potentially subgroups once we have done this enough times. And the variant burden is about that polygenic question that we were looking at a couple of slides ago. Now, flowing on from there, we look at the RNA and protein consequences, as Marcel was describing in those early slides. And we also look at ALS pathway identification. So lots of work has been done on ALS pathways in the last few years, in particular, where we're starting to develop a much better picture about the types of biological pathways that are always involved in ALS. It might not be identical components of those pathways in all patients that suffer from this disease, but those pathways are just, you can't avoid them when you're diving deeper and trying to understand the disease process. Now, from that, those outputs from our system, then we take things through to a testing phase of our DIGAP process. And here we're looking to identify potential disease mechanisms. This is where we have to take things from the dry lab to the wet lab. So we're basically testing our bioinformatic findings. So once we found these potential disease mechanisms from the computational steps, the validation takes place. Some parts of our validation process are really uniform, the things we always have to do. Sometimes they need to be quite bespoke though. We're working on standardizing them as much as we can. And from that, if these variants and biological pathways do come through that wet lab validation process, then we consider that we've identified new therapeutic targets. Next slide, please. This is just an overview, and I'm not going to go into too many details about this. Don't worry, I know it's a very complicated looking figure, but it's just an outline of our variant prioritization system, which I said is the part that's been the most useful to us in our research at the moment. So it starts with all of the variants coming in. Excuse me. And there are millions of variants in every single human being. There's lots and lots of variation amongst our species. Almost all of it doesn't have a significant biological consequence, or at least not a disease-relevant biological consequence. And we add our sort of special spice to the mix, if you like, our synergy score system, which is something that we had to work on in-house to make this prioritization work. The first thing then we do is cross-reference for known causative variants. That's a really easy thing to explain if we can find them. Like if we found a SOD1 A4V variant, for instance, that's one of the best described and oldest discoveries as these causative variants in ALS. But as we were alluding to earlier, that's actually pretty rare that we find anything even a bit like that, that has that level of statistical evidence supporting it. So then it goes through the novel variant prioritization pipeline. It looks like this pyramid. It starts at the top by finding new variants in old genes that we already know we need to be wary around. So that we'd be silly not to test that. But it does go down to sort of the tiers 4 and 5, where we're looking for genetic variants that they're very, very rare. They are expressed in the right tissue, so they're relevant to the brain and spinal cord. But they've not been described in ALS before, sometimes to any degree at all. And sometimes these genes just lack a good description, and that's where the non-standard component of our wet lab validation becomes very, very important, that we understand what's going on with that gene and what it might be doing. But it's relevant to, yes, the correct tissue, but also the pathways that I was talking about earlier, which is something we've invested a lot of effort in understanding. It helps us contextualize new genetic discoveries at the bottom of our pyramid, which is the bulk of what we find, and put them in the context of ALS in a way that it makes them actionable for those next research steps at the very least. There. Next slide, please. Cool. And this is just a little snapshot of what that tiered system looks like. So these are the results from one of those tiers from a study that we ran late last year and early into this year. So you can see here we've got some different types of genetic variants on the left-hand side of the top panel, and a number of genes that carry these extremely rare, predicted to be bad news by all of that thing that was a single bubble in the previous diagram, but I mentioned was actually very, very complex. So that's where that's all come from. And the panel on the bottom just shows some of the pathways that we're interested in looking at. So I'm sure individually, none of these would come as a surprise. They're things like, neuroinflammation and the organization of the synapses that communicate the messages from the brain down the spinal cord into the muscles. And we think this is useful because with 90% of sporadic patients having no family history, no genetic findings, with that sort of level of statistical support that we get from GWAS in particular, but occasionally pedigree studies as well, we know that we need to identify these novel genetic variants in most patients that we would ever look at. So that's why we see so much value in this system. And I'll show you now why a test case in which we were successful. Okay, next slide, please, Marcel. Cheers. And yeah, one through this as well. So this is a case study where we ran our complete pipeline on a particular sample in our research study that we've been, as I said, doing since sort of late last year. And this is what's called a de novo variant or mutation. So this is where the mother and the father, neither of them have this variation. And we know that because we sequenced the mother and the father of this patient as well, just to be sure. And we validated that it was definitely de novo using our wet lab validation component of the process, which was great. The computational side of things was really interesting as well. There was overlap with other neurodegenerative disease. All of the predictions in our, again, that little blue bubble, the complex one, said that this genetic variant is bad news. And it's not been seen in a human being before. It was really that rare. So we gathered up quite a significant amount of evidence behind this. And we thought, well, that's great. But before we go to the wet lab, let's have a look at some modeling. And that's on the next slide. So when we do find interesting cases like this, where it's a very new gene, it's a mutation that's not been seen before, we do invest a bit of extra effort to characterize it properly. And what we've got here is, you're looking at all these twists and bends. That's what the protein actually looks like, or at least our computed reality of what that protein actually looks like. And where this genetic variant occurred, there was some pretty big differences. So in the normal protein, that's this section that's kind of zipped closed really very tightly at the molecular level. It doesn't move very much. It is not open. But when we have the variant present, it's busted wide open. It's even letting water molecules get into this pocket of the protein where they shouldn't be. And that has high potential for being disruptive to that protein's structure and function. So this told us that we probably do need to glove up and get our hands dirty and take some measurements in a motor neuron cell line. That's on the next slide. Now, this is very, very complex data. I'm not going to describe it in detail, but what we're looking at is the electrical activity of the model motor neuron line. Now, this was really interesting because what this panel of results is telling us is that when that specific genetic variant is present in the model, it creates a hyperexcitable phenotype in those cells. Now, that really leapt out to us because hyperexcitability is strongly associated now with ALS, It's an early sign of the disease manifesting, and it's sustained over the course of the disease as well. And here we have this new variant and a new gene causing that specific phenotype. So that was really, really interesting to us. Next slide, thanks, Marcel. And we think that that's sort of our burning building, to use that analogy from earlier in the slide deck. So this is the case where we've got the protein finished, but it's on fire, unfortunately, and it spreads damage to nearby proteins and structures in the cell. So to summarize, we identified and characterized a unique genetic variant in an ALS patient. The variant is totally new to us and to the ALS field in general, and it drives this hyperexcitability phenotype. Next slide, please. So that opens some new possibilities for us, right? Now that we've tied it to a specific phenotype, we consider it as a therapeutic target, at least for that patient. And if we see any other variants in that gene again, perhaps they could go the same route. So we're currently developing some therapeutic strategies. This is what they look like. So drug screening to identify already safe and approved drugs that may act on this gene. That's sort of the lowest hanging fruit, if you like. We also have the capacity at GenieUs to develop new drugs that target this gene, which again, with the expectation that we would be making lots of new genetic discoveries, that's a good capacity to have. You might not have a molecule lying around that can target what you want to. We're also investigating supplementation with healthy RNAs encoding that gene of interest. So where we do have something that is damaged and has the capacity to spread that damage, supplementing with the normal version of that RNA may be a sensible strategy. So we are investigating that in the lab with the help actually of the guys that came up with the, I believe it was the Pfizer COVID vaccine. So it's a now very well tested technology that we know is quite safe. And of course, we're talking about antisense oligonucleotides as well. There's a very, very exciting sort of leap forward in this technology called allele selective antisense oligonucleotides. This is where you can simultaneously decrease the level of the wonky blueprint that's not doing us any favors and increase the expression of the protein from the one that works. So think of it like two blueprints, one of them's no good, one of them is correct. You can sort of throw away the incorrect one and only build from the correct blueprint. So yeah, next slide, please. So yeah, I'd just like to thank you all for listening. I hope it was interesting. And thanks very much to Gethin and the folks at MND Australia for inviting us along. That was much appreciated. And I'd like to thank obviously the team who is in the left-hand side. They're some of the best people I've ever worked with. And of course, this, sorry, it's a bit of a logo salad, but we have got a lot of people in our corner helping us now, and it just wouldn't be right to leave anybody out. So thank you to everybody involved with them as well. And of course, Marcel and I are happy to take any questions you guys might have too.
Thank you, Sam and Marcel. That's a fantastic example of how we can really take the genetics to the next stage with some, and the complexity needed as well. You know, it's not a simple, you know, okay, you've got the genetics, you've got the genes, but to really interpret it and get to the treatment stage, it needs next level technology. We're right at the back of the day, so I'm not sure if there's too many questions coming. We've lost a few people running off to other appointments, etc. Do we have any burning questions? If not, I think some of the GenieUs crew will be here tomorrow as well to answer questions as well. I'm not. No one's putting a hand up or putting anything in the chat at the moment. Just as a really brief wrap up, because we've got plenty more tomorrow. That was a huge amount of what I think was absolutely brilliant information, and I think everyone's going to come away from this knowing a lot more about MND ALS and the research behind it. One quick reminder though, it is Australian Eastern Standard Time, 9:30 tomorrow. So please try and get on there as early as you can. Richard or Terry, did you have anything else just to close off today? I mean, it must be late for you guys now, so it must be your bedtime.
Yes, it's been late, but it's been a great meeting. Learned some amazing things. I think you have a fantastic research portfolio there in Australia. And I just hope you can find a way to get these talented new research ambassadors involved.
I think so. I think we're seeing some really great connections already. So I really like the idea of the getting the sort of ambassador team together to interact with the researchers. I think that would be fantastic, and that's something we've been thinking about. Okay, that's just a real brief wrap up. Terry, did you want to add anything? Or...
I ditto for what Rick said. I think it's been a great evening it's evening for us, and and you know, I learned a lot. Genius is doing some great work.
Yeah, definitely are. Okay, so I'll let everyone go and have some lunch or whatever, afternoon tea now, and we'll see everyone who wants to be here tomorrow morning at 9:30. So thank you all, and see you tomorrow.
Thank you.
Thanks again.
Thanks, everyone. Bye.